Begin with a rapid review of microbiology. You know we've divided it into segments, that is general microbiology, immunology, bacterio, viro, fungus and parasitology, and that is exactly how we're going to proceed in that order. The first part of course is general microbiology, which gives you an overview of everything, but it's actually not just related to some general concepts, but it's actually going to include a lot more. That is, we are not only going to discuss all the microscopes, the scientists, the stains, but we are also going to go into chapters such as genetics. Bacterial genetics is something which we need to study. We are also going to study sterilization and disinfection, and we are also going to touch upon the culture media. So all of these, of course, I have spoken about in a lot of detail in your main videos, but I'll try my best to cover the most salient and important features for your exam as well. So let's get going. But the first thing, before we start with all of it, we must quickly talk about the scientists. What are the different scientists and who were they, how they contributed into all of microbiology? The first one whom we start with is said to be the father of microbiology, and that is Sir Louis Pasteur. If you remember his name Pasteur, you know that there are two things named after Pasteur. That includes what? Pasteur Institute in Paris and pasteurization of milk. Common sense, they are named after him. Other than that, we have L for Louis, and L for which liquid or which media did he provide us? So he's given us the liquid media, not the solid culture media that we use nowadays. Nowadays we use blood agar, chocolate agar. He didn't give us that. He gave us the L for L, that is liquid media. What else? So two things with Pasteur are done. One thing with Louis, L for L is done. Now we need to know he's the father of microbiology. So which means we are going to split the father word into multiple other alphabets. F is going to stand for the fermentation principle. He's given us the fermentation principle. AT is going to stand for autoclave. All of you study autoclave in sterilization, disinfection. He gave us autoclave. Then also when we're talking about father, and if I take these THER, he's related to a lot of theories. So he's told us that there is nothing known as theory of abiogenesis. He's disapproved it, and he's approved a theory called the germ cell theory. So next are the theories. So this is your final mnemonic that you need to follow for Sir Louis Pasteur. Finally, he's given us the very, very famous CAR vaccines. What are CAR vaccines? They are cholera vaccine, anthrax vaccine, and rabies vaccine. Repeating once again, not their organisms. He's given us the vaccines for these, right? So that is all about Sir Louis Pasteur. The CAR vaccines have been given because now we go on to a very modern name, Robert. Robert is sounding very modern. So now he becomes the father of modern microbiology. And please note, Robert Koch means everything regarding Koch that you will read is to do with him. So whether I ask you who gave us Koch postulates or who gave us the Koch bacillus, everything regarding Koch has been given to us. So I'll take the K and O of Koch, and Koch things are done. What about the C? Now the next alphabet C. He's given us three things with C, and those happen to be the cholera organism. Repeating once again, the cholera vaccine was given by Sir Louis Pasteur, but the cholera organism has been given by Sir Robert Koch. Then he's given us the solid culture media. Repeating once again, the liquid culture media was given by Sir Louis Pasteur. The solid culture media has been given by Sir Robert Koch, and then he's given us a color or a dye, which is the aniline dye. So firstly, with Koch, you had to learn Koch postulates and bacillus. With C, you had to learn cholera organism, the color and the solid culture media. And with H, he gave us the test for motility. He's given us hanging drop motility. So these are the contributions of Sir Robert Koch. Now, on that note, I'll quickly make you revise the Koch postulates also. What were these Koch postulates? Earlier, it was said that we have 4 Koch postulates. Now it is said that we have 5 Koch postulates. Let me first tell you these 4. What are the 4 old Koch postulates? Number 1, constant association with the disease. What is constant association with the disease? For example, if I say that there is a person who or for example, there is a bacteria called Mycobacterium TB. Tell me, what does Mycobacterium TB cause in us? Of course, ma'am, in every human, it is going to cause tuberculosis. Suddenly, it's not going to end up causing cholera, right? It's of course going to cause TB. So there's a constant association that whenever this organism will come, it will cause the same disease. It will always cause TB only. Suddenly, it's not going to cause gonorrhea or cholera all of a sudden, right? That is the first Koch postulate. Second Koch postulate says you should be able to isolate it in a culture media. So if I want to isolate the TB in a culture media or any other bacteria, like for TB, we have the LG media. Should you be able to culture it? Yes. Now, what is the third Koch postulate? Listen to that carefully. If I take a little bit of the colony from this culture media, if I take a little bit of the colony and I put it into some animal, the favorite lab animal that we have is a rat, it will cause the same lesion and the same disease in the rat also. So it should be able to cause the same lesion and the same disease in the animals also. And now from this animal, from this rat, if I try to re-isolate it, should I be able to grow it once again on a culture media? Yes, I should be. Just like you can grow it from humans, you should be able to grow it from animals also. So what are the 4 Koch postulates? Constant association, same organism, same disease always. You should be able to grow it in a culture media. You should be able to cause the same disease in animals, and you should be able to isolate it from the animals also. But now they say there is a 5th Koch postulate that whenever there is an organism or whenever there is an infection that occurs in any of us, tell me something. When we got infected, or many of us have gotten infected with streptococcus, strep throat, pharyngitis, do we develop antibodies? Yes. So this means we should be able to have antibodies in the serum. And that is the 5th Koch postulate that has been given, that whenever there is an antigen, you should be able to make antibodies against it. That makes the Koch postulates, which were given obviously by Sir Robert Koch. Let's move forward to the other scientists. Now, before I move forward, I just want to ask you, whenever I tell you any rules, there's nothing 100% in medicine. There will always be some exceptions. So which are the organisms that do not follow these Koch postulates? And they are LPG. What is LPG? L is going to stand for Mycobacterium lepra. Can you grow this in a culture media? No. This is only grown in the 9-banded armadillo. You cannot grow it in a culture media. What is P? P is going to be Treponema pallidum, and G is going to be gonorrhea. So remember, LPG do not follow the Koch postulates. That is your mnemonic. Let's move on to the other scientists. What did Sir Paul Ulrich give us? Common sense. Ulrich must have given us the organism Ulrichia, which was the 2022 exam question in INICET, Ulrichia organism. After that, what has Sir Paul given us? So number one, please remember, he is said to be the father of chemotherapy, right? So CH for chemotherapy. Next, he's also been given some awards. So P for prize. Where has he been given a prize? For toxin, antitoxin standardization. He told us that if there's a toxin, there will be an antitoxin. So he's given us that concept. He's got a prize. And A for, he's also given acid fast stain. Acid fast stain means now what you call as the ZN stain. Nowadays you call it ZN stain. But you know who originally discovered it? Sir Paul Ulrich discovered it. Let's move forward. What did Sir Joseph Lister give you? Joseph Lister. Joseph Lister is the father of antiseptic surgery. He told you that you have to maintain antisepsis environment in the OT. You have to use disinfectant, sterilization. So the father of antiseptic surgery is Joseph Lister. Anton von Leeuwenhoek, L for L. Which microscope did he give us? Light microscope. So Anton von Leeuwenhoek is going to be for light microscope. Means now you will ask me who is for electron microscope. Ernst Ruska. Ernst Ruska is going to be for electron. So simple. Leeuwenhoek, L for L, light microscope. Ernst Ruska, E for E, electron microscope. Edward Jenner. When I say Edward, this reminds me of the first vaccine. Edward Jenner gave us the first vaccine, and I think we all know the first vaccine was for what? It was for smallpox. Now smallpox has been eradicated after 1980, but the first vaccine was given by Sir Edward Jenner. What did Carrie B. Mullis give us? CABM, 3 alphabets, gave us PCR, polymerase chain reaction. So Carrie B. Mullis gave us polymerase chain reaction. What did H.C. Graham give us? No points for guessing. The Graham staining that you study was given by this scientist. Coming to the next one, what did Klenberger give you? Klenberger has given you the L forms, the cell wall deficient forms. The cell wall deficient forms or the L forms were given by Klenberger. What did Alexander Fleming give us? We have no points for guessing this. Alexander Fleming gave us penicillin, which is still being used till date as a drug. So this was first given, and this was a neat UG question also. Barbara McClintock. This is important because Barbara McClintock gave us transposons. For those who don't know what are transposons, they are referred to as jumping genes. Jumping genes means they can constantly change their location and function in the chromosome. So remember, transposons or jumping genes were given by Barbara McClintock. Now I want to tell you 2 Nobel Prizes which were given during that COVID era when we were in a lockdown, 2020, 2021. This is when it was given. So who and why were these 3 scientists given a Nobel Prize? Newton, Alter and Charles M. Rice. They were given this for their contribution of everything to do with hepatitis C virus. So remember, Newton, Alter and Charles M. Rice for hepatitis C virus. And the next Nobel Prize was given to, these are not doctors, these were 2 chemistry scientists, that is Madame Carpentier and Madame Jennifer. So Carpentier and Jennifer were given a Nobel Prize for their discovery on CRISPR-Cas9, which is nowadays being studied a lot. And I will also teach you under the bacterial genetics portion. So yes, that is what wraps up all of your scientists. I hope that is settled for all of you. Right after which we are going to start off now with the different stains and the microscopes that are needed. First, we'll go on to the basic stains on which they ask you a lot of questions in the exams, and these are the differential stains, and the word itself tells you difference. It's going to tell you a difference between, say, gram positive and gram negative, acid fast or non-acid fast. So some of the other difference will come up. Let us begin with the bread and butter of a microbiologist, which is a gram stain. And we all remember the very, very famous mnemonic for it, which is referred to as come in and stain. So what is CIAS, that is come in and stain? Number one, what we add is C for crystal violet. Then I is going to be for iodine. A is going to be for alcohol or acetone, anything with A. And S is going to be for safranin. So here it becomes in front of you, come in and stain. In other words, if I ask you which of these is going to be the primary stain, and you will say the primary stain is going to be crystal violet. Then if I ask you what is the mordant? Mordant is something which is going to fix the stain. It's a fixer or a mordant, and that is going to be iodine. Then what is the purpose of adding alcohol? That is going to be a decolorizer, alcohol or acetone. And finally, what is the secondary stain? And that is going to be the safranin. So crystal violet, iodine, alcohol and safranin. But which out of these is the most important step that we have? The most important step or the most crucial step that we have in this entire situation is going to be the decolorization, that is adding alcohol, because too much of adding of alcohol can result in excessive decoloration. Less of alcohol will result in less decoloration. So that is the step that a microbiologist has to monitor the most, and your end result is in front of you. The gram positive organisms, as you can see over here, are going to get a purple color because they are going to pick up all of the crystal violet and keep it till the end. But the gram negative organisms, as you can see over here, they are going to get a pinkish reddish color because they are going to get decolorized. They will give away initially they got colored with crystal violet, but they gave away all their color when I put alcohol or decolorizer, and then they picked up the red color from safranin. So remember, gram positive organisms are purple and gram negative organisms are pink or red. That is gram stain. But so I'll always say something is gram positive, something is gram negative. But is there something which is gram poor? Gram poor staining means neither can you put it under gram positive nor under gram negative. Yes, MRCS is gram poor. What is MRCS? MRCS stands for Mycoplasma, Rickettsia, Chlamydia and Spirochetes. The last chapters that you have, the 4 last chapters of bacteriology, which are categorized as this only. That is Mycoplasma, Rickettsia, Chlamydia, Spirochetes, MRCS, very famous surgery exam. That is something which is going to be poorly gram staining. So the gram stain is done. Shall we move on to the next one? Put a tick mark on gram stain and move on to the other one, that is the famous acid fast stain, which is famously also known as the ZN stain. Now Ziehl-Neelsen stain also has a mnemonic, which I hope all of you remember. What was the mnemonic for Ziehl-Neelsen stain? CHAM, that is class has asked mnemonic. Class has asked mnemonic. And I always tell students, this is a mnemonic which I once made impromptu in class, because student asked me, ma'am, iska bhi koi mnemonic? Tell us some mnemonic for this. So I said, okay, let me make a mnemonic right now. And this turned out to be a very impromptu 2-second mnemonic. That is, class has asked mnemonic for the Zn stain. So what is class has asked mnemonic? That's in front of you. So the first stain that you are adding, this reddish color stain, is carbonyl fuchsin. That is why you will realize all the organisms are coming out to be red in color, and that is what they ask you as well. What is the color of the organism? It's red in color. After that, class has, you're going to put heating. Heating is something which is going to act as a mordant over here. Remember what was the mordant that you had in gram stain? In gram stain, the mordant that you had was iodine. Whereas what is the mordant that you have over here? It is going to be heat. So first you have carbonyl fuchsin, then you have heat, then you add acid. Which acid am I talking about? Sulfuric acid. Sulfuric acid is added, and finally you add, can you see what is the color of the background? The color of this entire background is blue. Why is it blue? Because you have added a blue color stain that is known as methylene blue. So repeat with me: carbonyl fuchsin, then heat, then H2SO4, and finally there is methylene blue. So now let me tell you one more thing, guys. How much of H2SO4 do you have to add? That is also equally important. And what do I mean by that? For example, you are dealing with Mycobacterium TB case. So for TB, remember, you're going to go in for T20. You will go in for 20% H2SO4. Whereas what is for M. lepra? And see, lepra has how many alphabets? 5 alphabets. So for M. lepra, you are going to go in with a 5% H2SO4, and the same is applicable for the coccidian parasites also. Please note, when you use 5% H2SO4, that is known as the fight for Racco stain. That is referred to as the fight for Racco stain. So please remember, fight and 5, they sound similar, na? So remember, fight and 5 is the same thing. So fight for Racco stain is 5% H2SO4 used for 5 alphabet lepra bacillus. I can also use it for coccidian parasites. What is for the next one? Nocardia and Legionella. We use 1% H2SO4, and what do we use for the spores and the head of the sperm? How do they, how do they write it down? Spores and head of the sperm, SS. So it is going to be 0.25 to 0.5% H2SO4 is going to be used in this scenario. So here you go. That is the Ziehl-Neelsen stain, the different concentrations of H2SO4. This is a case of TB. So what do you think? How much of H2SO4 would I have used? T for T, 20. Actually, in many textbooks, rather than 20, they write 20 to 25%, and that is acceptable. Now, is TB the only thing which is acid fast? Of course not. There is a very, very long mnemonic. My nose is cold and hot. These are all the acid fast things. And this mnemonic is just not a random mnemonic. I'll tell you a logic behind it. But what is my nose is cold and hot? So my, of course, stands for everything mycobacteria. Typical mycobacteria, atypical mycobacteria, everything is acid fast. No for Nocardia and Legionella. I, look at the I guys over here. So I is going to be, can you see these spora brothers? Isospora, cyclospora, and my nose is cold. C for Cryptosporidium. So what is this together known as? If I say Isospora, Cyclospora, Cryptosporidium, which family am I talking about? I am talking about the coccidian parasites. Yes, I just told you coccidian parasites are acid fast, but I'm missing out on one alphabet. My nose is, S is going to be for, I just now told you, spores and head of the sperms. They are also going to be acid fast. So my nose is cold. And finally, hot. What do I mean by hot? Hot refers to, you're going to split it. You're going to split it into HO and T. So what do I mean by that? HO stands for hooklet. Hooklet of hydrated. Everyone knows hydrated cyst. Hooklet is acid fast. And T is going to be tinea. Tinea ka egg. Ma'am, you will ask me, ma'am, tinea kaun sa tinea? Tinea saginata, tinea solium. Go for the T. Tinea saginata egg. That is why I said hot T. T. Tinea saginata egg is going to be acid fast. So my nose is cold and hot. These are all the acid fast things. What is the logic behind this mnemonic? Why did you use this? My nose is cold and hot. What is cold and hot happening in Zn stain? So look at this, guys. Right now, did you say that there is a heating step? Heating wala step hota hai in Zn stain, agree? If I cut out this heating step, I don't heat. I do the first, third, and fourth step. I don't do the heating step. What is now that going to be known as? So please note that if I do not heat, can I call it cold? Yes. So class has asked mnemonic means carbonyl fuchsin, no heating, acid H2SO4 and methylene blue. That means when there is no heat taking place, it is a cold Zn stain. Cold Zn stain is known as the Kinyon stain and is used for the coccidian parasites. Can you all hear the KK sounds coming your way? We have the Kinyon stain, which is a cold Zn stain, which is used for the coccidian parasites. If you go in for the advanced exam questions, this is given one more name, and that is known as the Gabbett stain. So yes, all in all, these were all the variations that you had to know for the Zn stain, which is also done. We move on to the last one, and that is the Albert staining. I think everyone knows Albert staining is something which you use for which organism? Which is the organism that I've drawn over here? This organism is going to be Corynebacterium diphtheria. So I can say Corynebacterium diphtheria is a gram-positive organism. Why do I need Albert stain for it? Because I don't want to see Corynebacterium diphtheria. I want to see something that is there in Corynebacterium diphtheria, and these are these granules. What are the names of these granules? Do you all remember? The names of these granules are the volutin granules. These purple color granules are volutin granules known as the Babes-Ernst granules. So ये तो fancy नाम है. You will have to mug it up. Volutin granules, Babes-Ernst granules. But what is their logical name? Bipolar and metachromatic. What is bipolar? See, they are present at the two poles. They are always present at the two poles. So bipolar and metachromatic is what you have to know. So what is this Albert stain? I learned it as TIM, but there are two ways of learning this stain, right? There are two ways. The textbook says there is an Albert 1 solution and an Albert 2 solution. Albert 1 and Albert 2 में everything is present in the Albert 1 solution. So out of T for toluidine blue, M for malachite green, and a little bit of acetic acid. Everything is present in Albert 1 solution. Albert 2 solution only has iodine. So if they ask you, what will you add first and what will you add second? So common sense tells me that Albert stain 1 I'll add first and Albert stain 2 I will add next. And that is going to be, if they ask me, don't tell me in terms of this 1 and 2, tell me the three chemicals in it. You will say, okay, toluidine blue, iodine, and malachite green. So what did I say? Toluidine blue, तो कुछ तो blue color है. Malachite green, कुछ तो green color है. There is a blue color, there is a green color involved in the story. Yes. Can I see the green color? Yes. The organism has stained green in color. So malachite green is going to give a green color to the organism. And toluidine blue You say, ma'am, I can see that it has to stain the voluten granules. So, ma'am, toluidine blue should have given a blue color to the voluten granules, but here it has given a purple color. What is this? I am saying that I'm going to flunk the exam, but on the day of the exam, I top the exam. Am I being two-faced? Yes. Do we all have that topper in our class who says I'm going to fail, but is topping the next day? Same way, this stain is saying that I have a blue color, but on the day of the exam, the color that it's giving is a purple color. What is this stain known as? This is known as a metachromatic stain. Has a different color, will give a different color. So if I give you a bottle of toluidine blue, you'll say, okay, ma'am, it's a blue color stain. But when you look at it under the microscope, it will start looking purple. Metachromatic, the two-faced person. That is why can I say voluten granules are also known as metachromatic granules. Please remember, toluidine blue is an example of a metachromatic stain. The two-faced topper of your class is how you will remember it. Yes, I hope everyone's clear with it. So finally, they'll give you the same picture which comes in the paper. They will say, okay, what is the reason for the green color bacillus? The green color is because of malachite green. And what is the reason for these purple color granules? That is toluidine blue, Albert stain for Corynebacterium diphtheria. Now they will also ask you, is this the only stain that you have for voluten granules? No. For voluten granules, there are a lot of stains. I have made a plan. We are making a plan. We are going to make a plan for the voluten granules. What is the plan? Out of plan, I know anyway Albert stain to pad liya hai, right? Other ones are P for Ponder stain, L for Loeffler's methylene blue, which is now coming across as the best stain and a famous PYQ. And finally, you have the Neisser stain. So please note, Ponder's, Loeffler's methylene blue, Albert and Neisser, the plan. We are making a plan for the voluten granules. That is what you had to know. So this is also over. So you've done what all? You have finished off your Gram stain, Zn stain, Albert stain. What are some miscellaneous stains that you have to know? Okay, so now let's talk about Cryptococcus. Please note, for Cryptococcus, although it's not a bacteria, because till now I was only making you focus on bacteria, Cryptococcus is a fungus, but we all know that for Cryptococcus, there is a very famous stain which is known as the negative stain. And some examples of negative stain that I want to give you is number one, India ink stain, and the second one is going to be nigrosin. India ink and nigrosin are the two stains that you have for Cryptococcus. I'll give you an example. Just focus on the first picture for the time being. What is this? This is an example of a negative stain. What do I mean by negative? That the background is going to take the stain, not the organism. Can you see? The capsulated organism is automatically highlighted. Capsulated organism is automatically highlighted. So for example, if I, like you can see, all of you can see the prep ladder edition has a black background. So anything that I write with white automatically gets highlighted, right? Similarly, I have stained the background in this scenario. So the organism automatically gets highlighted. That is a negative stain. What is the stain that you have for flagella and spirochetes? Basically, I want to pick up organisms which are very thin organisms. Out of them, please note that especially for, so before that, I think most of you have read your main videos. When I want to stain very thin things, what are the kind of microscopies that I use? Dark field microscope. I will be teaching this to you in exactly 5 minutes. Dark field microscopy. But you say, ma'am, I'm not that rich. I don't have a dark field microscope in my lab. What am I supposed to do? Can you tell me that using the same light microscope, what are the things that I can use? Yes, light microscope ke saath you can use silver stains. You can use some silver stains. I'll give you some examples. There is for spirochetes, there is something known as a Fontana silver stain. There is something known as a Levadity silver stain. These are stains that you have for spirochetes, which I have taught you in the spirochete chapter also. What about flagella? For flagella, I've got you separately. Flagella is going to be, because you can see flagella has a lot of L's in it. Flagellar stain is going to be the Leifson and Rieu stain. So flagellar is going to be Leifson and Rieu stain. Coming to the last one, spores. You'll say, why are we studying spores, ma'am? Number one, spores are Zn stain positive. Right now only you told me, but is there any other stain for spores? Yes, S for S, and that is known as the Schaffer and the Fulton stain. Please get used to these fancy terms. For Cryptococcus, it was negative stain like India ink and nigrosin. For thin structures, we prefer a dark field microscope. If not that, we will go for other stains. Like for spirochetes, we have S for S. We have some silver stains that includes the Fontana and Levadity. So we'll have these silver stains put up our way. For flagellar, LLR, that is Leifson and Rieu stain. What do we have for spores? One is Zn stain and the other one is S for S, and that is Schaffer and Fulton. Do you want to see the Schaffer and Fulton stain? Yes. The Schaffer and Fulton stain is known as the modified Ashby stain. S for S, repeating once again. What is a spore? Spore is the sleeping stage. Sleeping basically means the dormant stage that you have for a bacteria. The dormant stage that you have for a bacteria. And that basically means whenever you, you must, all of us have been through this phase that whenever we're like totally idle, we are not studying, we're not doing anything, we're not doing anything productive in our life and only sleeping. If you sleep till like and wake up 2 in the afternoon, 12 in the midday, you will always get this taunt from one or the other family member or your parents saying that, oh, the king or the queen has finally woken up. Miss so-and-so has woken up. Mr. So-and-so has woken up. So that is why I refer to it as this. So because spore is always the dormant or the sleeping stage, I call it the Miss spore. Miss so-and-so has woken up. So what is MS? M means that one of the stains that is being used over here is malachite green, and you can see that. You can very well see that the spore has come out to be green in color, malachite green. And then what is the color of the organism? The organism is going to get a saffron color over here. So I can see that, yes, there is a reddish color. So it's simple. We have the malachite green and the saffron in red. So Miss spore, malachite green, saffron in red. These are the two stains that I'm using for spores. And this is also referred to as the famous modified Ashby stain. And that definitely wraps up all of these special stains also that you had to finish. What do we move on on to next. We start now with the next but a very important topic, not only for micro but also for path, and that is microscopes, because this is what the entire two subjects revolves around. And you know, the first one that I have to begin with is the very famous light microscope. Now, when I'm talking about the light microscope, the first part of it that I have to label is the source of light. So the question that you get in your previous years is, what exactly is the light that is being used over here? So remember, we are talking about the transmitted light, and a very simple way of learning it is that in the light microscope, it ends with a T, so the type of light is the transmitted light. The light can go in any direction. As you all see any bulb or tube light in your room, the light can go in any direction. I want someone to narrow it down, someone to condense it. So what you have over here is referred to as the condenser. And this condenser, inside it has an iris. Like you have an iris in the eyes to bring the light to a direction. Same way the condenser has an iris inside it because it wants that the light should come over here. What is this square thing known as? This is known as the stage. Why is it called the stage? Stage always has the hero performing on it. So the stage is going to have the slide. Whatever glass slide you have is going to be kept on the stage. Now after that, let's come to the lens system. So this is the set of lenses which is near the slide, near the object. So these are said to be the objective lenses. Similarly, we have another set of lenses, but that is from where you are looking. This is near your eyes. So what kind of lenses do these become? These become the eyepiece lens. So we have an objective lens and we have an eyepiece lens. And next what they will be asking you is, what are the magnifications of these? So objective lenses, as you saw, there were many. I can see 4 objective lenses over here. Eyepiece lens is just one lens. So the magnification of the eyepiece lens is always going to be 10x, right? Simple. One lens, 10x. But objective lenses can be scanner, that is 4x, low power, that is 10x, high power, that is 40x, and oil immersion, that is 100x. So remember, 4, 10, 40, 100, 4, 10, 40, 100. These are the 4 magnifications that it provides. That is why when someone is asking you what is the maximum magnification that you can get in a light microscope, you will always multiply objective into eyepiece. Objective into eyepiece. So eyepiece is always going to be 10x. There is no other lens available. For objective, you will have to ask the examiner, what are you using? Scanner, low power, high power, or oil immersion? So if they ask you what is the maximum possible is oil immersion, means 100x. Now multiply the two, multiply 110. What is the maximum magnification possible? It is 1000x magnification, and that's a very famous previous year question. What is the maximum magnification that you can obtain from a light microscope? Is 1000x. Okay, now on the same lines, I go on to the next one, which is a dark field microscope. And over here, you look at the alphabet R because the kind of light that is used is the reflected light. So you can see that the background is going to be very, very dark. Reflected light will make the background very, very dark. So you will obviously ask me, what is the use of this kind of a microscope? Whenever you have to look at something very, very thin structures are involved. Like, can you identify this bacteria, the spiral looking thin bacteria? It is the spirochetes, or anything else in a bacteria which is very, very thin, and that's going to be a flagella. So these two things are very thin. You can't see them regularly under a light microscope. No, you won't be able to see it here. So what are you going to do? You're going to look at it under a dark field microscope. Dark field microscope uses reflected light for spirochetes and flagella. Let's move on to the next one. This is important not for FMG students, only for INI-CT aspirants it is important. That is interference contrast microscope. You are trying to interfere. You're using this microscope, you're creating a contrast. When you're trying to interfere means when I say someone's trying to interfere in your life, he's coming and trying to know the details of your life. So whenever you want to know something about the inner details, the organelles, you are trying to interfere in a cell. That is interference contrast microscope. It is trying to know every chemical constituent, every little detail, whether lipids, proteins, nucleic acids. So simple to learn. When there is a microscope which can know every little detail of the chemicals, of the lipids, of the organelles, that is an interference contrast microscope, important for the INI-CT exam. Moving on to the next and examiner's favorite, and that is the fluorescence microscope, which has a U in it. That is why the kind of light source that I will be using over here will be an ultraviolet light. So now just imagine, if I ask you what is the principle of a fluorescent microscope, you will say there is an ultraviolet light. That ultraviolet light has gone and it has first gotten excited. There is something called an excitation filter. So they will ask you these questions: which microscope uses an ultraviolet light, which microscope has excitation and emission filters, and which microscope has a dichroic mirror. If you ask me the two famous questions of fluorescence microscope, what is the source of light? Ultraviolet light. What is going to be the mirror used? Dichroic mirror. And what happens is that initially the light that is coming is a shorter wavelength light, but there's something exciting it. So that shorter wavelength light will now be converted into a longer wavelength light, and that is the principle of a fluorescence microscope. The principle says that a light of a shorter wavelength will be converted to a light of a longer wavelength. And now you will ask me that, okay ma'am, I've understood the principle, I've understood the light, I've understood the mirror used, but what are those green color because of this fluorescence that you're talking about? Dye kaun sa hai? What color have you used? So remember, if you're wanting to look out for TB, there are two fluorescent dyes which are referred to as auramine and rhodamine. Auramine and rhodamine are the two fluorescent dyes that are present. Now, I hope you remember I'm talking about rhodamine and I'm not talking about rhodanine, and I'm taking you into pathology because rhodanine was a stain for copper. Here I'm talking about auramine and rhodamine. M, because remember M is for Mycobacterium TB, right? Okay, what is for malaria? For malaria, we have something known as acrid in orange, which I taught you in the malaria chapter also. What is it for fungus? Again, something that I taught you in the fungus chapter. It is CFW, that is calcoflower white. It is calcoflower white. And now I want to integrate pathology also over here. So now tell me, what is the fluorescent dye that you use in flow cytometry? In flow cytometry, what we use is FITC. What is FITC? It is fluoroisothiocyanate. Fluoroisothiocyanate is the dye that is used, FITC. So repeating, MTB, auramine, rhodamine. Malaria ke liye acrid in orange. orange, calcoflower for fungus, calcoflower, and flow cytometry के लिए fluoroisothiocyanate. Principle, shorter wavelength to longer wavelength. What is the light? Ultraviolet light. What is the mirror used? Dichroic mirror is used. But now they will ask you, are there any molecules which are autofluorescent? Means you've not added any color, you've not added any auramine, rhodamine, calcoflower, right? कुछ नहीं add करा, they will give a green color on their own. Autofluorescence, they will shine on their own. So please remember the spora brothers. This means the cyclospora and isospora is autofluorescent. If you ask me, ma'am, anything in pathology that is autofluorescent, and that is formalin. Formalin can shine on its own. And anything in biochemistry that is autofluorescent, it is NADPH. Chapters are different, but ultimately everything is related to medical science. So remember, the spora brothers will shine on their own. Then formalin will be fluorescent, and NADPH is also going to shine on their own. These are all auto shining things that you have. You don't have to add any stain. Okay, now on that note, we have one more microscope that you have, which is known as a phase contrast microscope. So phase contrast, what did you analyze? Difference in phases means that if you want to look at the internal details of a cell, or you want to see, I'll give you an example. Do you remember in pathology you study something called hairy cell leukemia, in which there are cells which are having a lot of hair, right? So hairy cell leukemia is related, you should be knowing that there is something where you want to see these hair. They have a different refractive index, and then the cell has a different refractive index. So basically when there are different refractive indices that are involved, that is when contrast banega, na? That is when I'll use a phase contrast microscope. So you can see over here that how will this be different from any normal microscope? Because I can see something known as a condenser annulus that has been placed over here. Some different ring has been placed in front of the condenser, and there is a phase plate that has been added. These are extra things, ma'am, that I can see. What? I can see an annular diaphragm or a kind of a annulus, a ring that has been placed ahead of the condenser, and I can see a phase plate that has been added. This will what will make me help, or will help me in maintaining that refractive index change, and will help me in knowing the internal details, right? Let's move on to the next one. What is this microscope? The microscope that looks like a spaceship. I always say the microscope that looks like a spaceship is electron microscope. So obviously the source over here is not any light. The source over here is electrons, and if you have electrons, you can't keep them in the air. You will have to create a module, or you'll have to create a vacuum environment. So can you see they've closed it all around? Otherwise electrons will all get lost. Now you have to keep them in a closed chamber, in a vacuum. So the source is going to be the electrons, and the medium is going to be the vacuum that is over there. Now let's know the differences. What are the differences between an electron microscope and a light microscope? Things that I've taught you in pathology also. So first and foremost, for the fixation, for electron microscope 2 to 2.5% glutaraldehyde, and for light microscope we all remember 10% NBF. For those who are not aware, NBF stands for neutral buffered formalin. 10% NBF is what is used. Next, where will you keep the tissue? Like let's go into a pathology example. You have a kidney biopsy. If a kidney biopsy has to be sent for light microscope, I'm going to keep it in wax. Whereas if a kidney biopsy has to be sent for electron microscope, I'm going to keep it in resin. What is the source? Of course, in light microscope, the source is going to be light. Light microscope, ma'am, common sense, it's transmitted light. Whereas in electron microscope, it's going to be electrons. So electrons ke liye, the medium that I will have to create is vacuum. Whereas for light can travel easily in air, so it's not going to be a problem. Coming to the slide, we all know that when I'm using a light microscope, the examiner is going to give me a slide which is made up of glass. Whereas when I'm talking about an electron microscope, the slide is going to be made of metal, and the metal which is most commonly used is copper. Which out of them do you think is going to give you a higher resolution? Of course, the higher resolution will be obtained by an electron microscope. This is common sense. But do you know how many types of electron microscopes? This is just one picture, but do you know how many types of electron microscopes that we have? We have something called scanning electron microscope and transmission electron microscope, SEM and TEM. It's very difficult to learn. So I always tell the students, learn from a day-to-day example so that you can recall and remember it later on also. So just imagine you are studying. So for example, if you were doing your first reading, you would have read all your main ideal situation. You would have read all your main notes. You would have done all your question bank, your textbooks, subject-wise books. Means you would have gone into every little detail. You would have transmitted through the books. You would have gone through every detail. So if you're using transmission electrons, means you're going through and through, you are actually going to get a two-dimensional view. Right now, you're not integrating it with any subject. You're picking up one subject-wise book, only reading that subject through and through. So you're getting a two-dimensional view. Is it going to take time? Of course, ma'am, one subject is 80 hours of video lecture and whatnot. It's going to take a lot of time. So I will cover very less syllabus in the same time because I'm going into every little detail. And the details, of course, will be very, very proper, detailed concept, foundation, internal details milega. Whereas if I am just 1 month away from the exam, and that is why you're listening to this video, that is the rapid review. If you're just 1 month away or 2 months away from the exam, very close to the exam, you will scan through. Quickly, you will flip through pages. You will say, ma'am, 15 hours, 20 hours, finish off the subject. I don't have time for 80 hours. This means, and at the same time, you will say, ma'am, integrate with medicine also, integrate with path, micro, सब कुछ integrate करो. Means you're wanting me to scatter things around. What electrons are we using? Scattered electrons. And what kind of a view will I give you? 3D view of all the subjects integrated together. And this means I will make you finish your syllabus very, very quickly in 15 hours. That means more sample will be viewed. But details, will I tell you like every little textbook detail? No. Details are not going to be there. All the surface important details I will tell you. So the surface details will be seen. That is how you learn scanning versus transmission. Scanning is your what? If I had to tell you, transmission electron microscope is full videos की तरह, and scanning electron microscope is rapid review की तरह. So transmission, when you're looking at full videos, you're going through and through, you will get all the details. You're only going to get that subject view, so 2D view, but you are going to take a lot of time in covering this. Versus if you're doing rapid review, I will integrate it with many, many subjects, a 3D view. I will make you learn it very, very quickly, and I will give you all the superficial details that you need to know. That is how you learn the different types of electron microscopes, which wraps up the story of microscopes also. What are the next topic that we start with now in micro, general micro, the anatomy and the physiology of the different bacteria. So let's begin with the next part, and that is bacterial anatomy, and you all know that I'm going to describe the entire bacterial structure to you. So of course, what we have in the outermost part is some of the bacteria have it and some don't, and that is a capsule. There's something known as a capsule, whereas other bacteria might have a little looser version of a capsule, and that is a slime layer. So either a slime layer or a capsule. If you ask me the difference between the two, remember, capsule is more rigid. It is more well demarcated, whereas slime layer, on the other hand, is very, very loose. As I said again, either of the two will be present. But is there any bacteria which has both capsule as well as slime layer? Yes, there is a bacteria which is known as Streptococcus salivarius. Streptococcus salivarius has both a capsule as well as a slime layer. So that's the first thing. But then you will ask me, tell me the organisms which are capsulated. In the main videos, I have given you a mnemonic called pretty nice capsules. What is pretty and nice capsules? All the organisms which you have read in bacteriology or elsewhere with P means which Streptococcus, not the Streptococcus agalacti, no, Streptococcus P wala, pneumonia. Which Klebsiella, P wala, Klebsiella pneumonia. Which Bordetella, Bordetella pertussis. Which Vibrio, Vibrio cholera, Vibrio alginolyticus, no, the P wala, that is Vibrio parahemolyticus. Which Clostridium, again, P wala, perfringens. Which Yersinia, P wala, that is pestis. So remember, P for pretty, all the P's that you have everywhere in bacteriology. Now coming to pretty and nice, what is N going to be for? N is for Neisseria meningitidis. So nice as a whole only sounds like Neisseria. Neisseria meningitidis definitely has a capsule. Then we have I. The I is going to be influenza. Of course, Haemophilus influenza. And pretty nice capsules. So C stands for the very famous fungus, which is Cryptococcus. And A stands for Staphylococcus aureus and Bacillus anthracis. Now, pretty nice capsules you have learned, but what are the exceptions? You know, all or most of these capsules are made up of what? Most of the capsules are made up of polysaccharides. So remember, all capsules are made up of polysaccharides except Yersinia pestis. I see peptide written in front of it. And Bacillus anthracis. I see a polypeptide written in front of it. So these are the two which are going to be made up of polypeptide, whereas all the other capsules are made up of polysaccharides. Okay, which of these is actually, you might have never heard. Have you ever heard that Staphylococcus aureus has a capsule? Even I have not taught you in the bacteriology segment. Why? Because the kind of capsule that it has is a very tiny one. It's a micro capsule that they are talking about. So that's a very rare INI CET level question that I would expect. The INI CET of 2023 had a question where Yersinia and where Neisseria meningitidis was asked as a capsulated organism. So this list is definitely important. Repeating, all the capsules are made up of polysaccharides except Yersinia and anthrax, which are made up of polypeptide. Okay, having discussed it, the next layer that I'm going to go on to is going to be, of course, the very, very famous cell wall. And you know you have to differentiate between a gram-positive and a gram-negative cell wall. And I always tell people that the best way of differentiating it is, remember the mnemonic PPPT. What is PPPT? That when I'm talking about a gram-positive organism, something that is going to be predominantly present is going to be peptidoglycan. It is predominantly made up of peptidoglycan. The content of peptidoglycan is too much, which you can see also over here. It's a very thick peptidoglycan layer. What is the T? It is also made up of or going to contain something called techoic acid. You can see molecules of techoic acid which are going through and through. What is the function of techoic acid? I've given you a mnemonic. Techoic acid is for ticna. Ticna basically means that this is something which is used for attachment. What else? What is this last P that I'm left with? Remember, all the gram-positive ones have something known as a pentapeptide bridge. Pentapeptide. What is pentapeptide? 5 peptides cross bridge that is going to be indicative of a gram-positive organism. Let me show you. So look at this. This is the difference between a gram-positive versus a gram-negative organism, and you can very well see that whether it is gram-positive or gram-negative, they are made up of NAM, NAG, NAM, NAG. So that's the rule that we have. NAM and NAG are molecules present in gram-positive also. NAM and NAG are molecules present in gram-negative also. NAM, NAG is cell wall. Someone asks you, what is the definition of cell wall? You will just say, okay, the formula of a cell wall is NAM, NAG, NAM, NAG. But do you see something which is a little different? If you see very carefully, pick up the NAM molecules, you will realize that these NAM molecules are connected by 1, 2, 3, 4, 5. The NAM molecules are connected by a pentapeptide bridge, but only in the gram-positive organisms. If I try to join and see, do you see any of those 5 molecules over here? No, here there is a khali line, there's an empty line. I don't see those. So simple, a gram-positive organism will have the pentapeptide cross bridge, it will have a lot of peptidoglycan, it will have a lot of techoic acid. Means a gram-negative organism will not have techoic acid, will not have this pentapeptide cross bridge, and peptidoglycan is very, very less. So hai kya? What does it have? It predominantly, if you look very, very carefully, it is made up of something known as gram-negative organisms have lipopolysaccharides. And what is lipopolysaccharides? Lipopolysaccharides is going to be made up of OPA. Lipopolysaccharide has region 1, region 2 and region 3, and be very common sense lagao. O to is OPA matlab region 1 is made up of O antigen. O for O antigen. If you're saying lipopolysaccharide, there has to be a lipo component and there has to be a polysaccharide component. So what do you think the P is going to be? The P is going to be the polysaccharide component. And what do you think A is going to be? That is the lipo component, that is lipid A. So simple. Why do I say region 1, 2 and 3 is OPA? Because now see with me. O stands for what? It is made up of O antigen. Then P stands for polysaccharide and A stands for lipid A. So OPA. OPA is the region 1, 2 and 3. And so now tell me, did you see much of peptidoglycan? No, because this is gram negative. So can you fill in the blanks for me? If I ask you who has predominant amount of peptidoglycan, you will say, of course, ma'am, too much of peptidoglycan is in gram positive and the amount of peptidoglycan in gram negative is little, because predominant amount of gram negative is made up of lipopolysaccharides. Lipopolysaccharides are present. So lipid content kisme zyada hai? Of course, gram negative has more of lipid content, gram positive has less of lipid content. Okay, NAM, NAG to dono mein hai, right? But who has a pentapeptide cross bridge? The pentapeptide cross bridge is going to be present in gram positive and not in gram negative. Who has techoic acid? Gram positive, not in gram negative. That's why I told you learn the mnemonic PPPT. P for gram positive, positive, P for peptidoglycan, P for pentapeptide cross bridge, and T for teichoic acid. After this, whatever is mentioned, if they ask you who has an outer membrane, gram negative, you will have to give something. Now, PPPT you have given to gram positive. So outer membrane, if you can look at this picture, outer membrane is going to be present in gram negative organisms who has a periplasmic space. This tiny little space, periplasmic space, is also present in the gram-negative organism. So these are all features of the gram-negatives. What is the question on periplasmic space? Can you recall something, guys? Periplasmic space has something hiding inside it. That is endoflagella. And endoflagella is a feature of which bacteria? It's a feature of spirochetes. Endoflagella matlab flagella will be hiding here only. It will not be coming out of the bacteria like this. It will be hiding inside. That endoflagella feature of periplasmic space is a feature that you see with spirochetes. Very, very important. So this is your entire bacterial anatomy that you had to know. If I come back now, you can very well say that, ma'am, in this diagram that you had drawn in the beginning, you had drawn this as the cell wall, then you've drawn a dotted diagram. Maybe that's the outer membrane. Achha, now I'm saying if outer membrane is drawn, is it a gram positive or gram negative cell wall? Of course, it's a gram negative cell wall. What after that? I can see then there's this reddish color thing which is drawn, and this is referred to as the cell membrane. Finally, we have the cell membrane that is present. Guys, can you see the cell membrane is taking two depressions like this? What is that depression known as? That depression is referred to as mesosomes or chondroid. Repeating once again, देखो outer क्या था? Capsule. Then ma'am, ये capsule था, then cell wall, then outer membrane. Finally, I have the cell membrane. And invagination, question number 1. Invagination of the cell membrane is going to be known as mesosomes and chondroids. क्या होता है? What are mesosomes and chondroids? They function as a respiratory unit. This is how the bacteria breathes. This is a respiratory unit, and it is more prominent in gram positive. Means it is present in gram positive and gram negative organisms, but it is more commonly a feature of gram negative organisms. So there are 3 questions on mesosomes or chondroids that you get. Question number 1: it is an invagination of which layer? The cell membrane. Which bacteria has more of it? Gram positive has more of it. And what is its function? It is going to be utilized as a respiratory unit. So we all have understood what are the mesosomes or the chondroids. What else is there inside this same one? You will now ask me that, ma'am, inside this bacteria, can you have some ribosomes? Yes, we have 70 S ribosomes that are there. Can you have a nucleus? Yes, but what is characteristic about the nucleus is you are going to have a circular nucleus. Matlab DNA. Bacteria also has to have DNA, right? So if I ask you what ribosomes are there, 70 S ribosomes are there. If I ask you what nucleus is there, as I had just drawn for you, it is a single circular nucleus that is present. These are all the parts of the bacterial anatomy. Coming back, I can see 2 projections coming out. One long projection like this, which is referred to as a flagella, and 2-3 tiny projections which are referred to as pili or fimbriae. So now we need to know what is flagella and what is fimbriae. Dono projections hai, but their functions are very, very different. So if I ask you what is a flagella, this is how a flagella is. Whether it is a gram positive organism or a gram negative or gram positive organism, I can see a flagella has a basal body. Right? Then it has something which is going to hook out like a hook. So I call it hook. And then it has a portion hanging, and that is filament. Everyone, what are the 2-3 parts of the flagella? Ma'am, base part, of course, basal body has to be there. So at the base, there is basal body. The one that is going to look like a hook, that has to be named as hook. And the one that is projecting out is known as the filament. So how will I know? Because gram negative and positive, both of them have. So I've kept a selfish mnemonic as MS, Miss Preeti. What is Miss Preeti? So M and S are the 2 rings, the basal body having only 2 rings. Having only 2 rings over here, that is, so means, now I can hide. Means, ma'am, these rings won't be there. If you're talking about a gram positive organism, you will see just 2 rings, and that is M and S, that makes it a gram positive. But if you see 4 rings, M, S, P, L, if you see 4 rings, that is going to make it a gram negative. So simple, M and S dikh raha hai, 2 rings dikh raha hai, that is going to be gram positive. That is what I kept as a selfish mnemonic. Okay, this was about flagella. What is flagella going to help in? Number 1, it is going to help in locomotion. Anything else? Does flagella also carry an antigen? Yes, flagella is antigenic. What is the name of the antigen? The name of the antigen is H antigen, which makes me want you, to answer a question. I'm going to make a tiny little table over here and ask you, do you all remember the Vidal test? Correlation. Vidal test uses, yes, for what? For typhoid. Very good. Vidal test that you use for typhoid utilizes 2 antigens, O antigen and H antigen. O antigen right now you have seen in the cell wall. Remember, the cell wall of a gram negative organism has OPA, O antigen. So O antigen is going to be present in the cell wall, and H antigen, right now I'm showing you that H antigen is nothing but the other name of a flagella that you have. So all your questions are done. Vidal test is done for you. But what is it helping in? It is primarily helping in movement. So your next question will be, where is it located? What all movements are there? So where is it located? See, one flagella, one end. Har jagah nahi hai. This is a bacteria, for example, and you have one flagella at one end. Why did I say that? Because 1 is sounding like vibrio. 1 is sounding like vibrio. Vibrio is 1, monotrichous, 1 flagella at one end. Similarly, pseudomonas. Pseudomonas I have learned as pseudomonoas. Pseudomonoas will tell me mono, 1 flagella, one end. So remember, 1 flagella at one end is present in vibrio and pseudomonoas, right? Then amphitrichus kya hota hai? 1 flagella is present on one pole, 1 flagella is present on the other pole. What is lopho? Lopho means tuft of flagella. You can see one bunch of flagella. So remember, tuft or bunch. Tuft or bunch is present in the bacters, Helicobacter, Campylobacter. So remember, tuft or bunch is going to be present in the bacters. And finally, peritrichus, the one that is roaming all around. When I say peritrichus, The one that I say peritrichous, number one, P for we have Proteus, or you can combine the P and R so that you don't confuse it with any other P. So P and R for Proteus. E for the remaining E is going to be for E. coli. And finally, instead of peritrichous, I'm going to add an L over here. And now the answer to the last one becomes Listeria. So which are the ones which have peritrichous? Number one, there is Proteus. Number two, there is E. coli. And then there is Listeria. Let's do a recap. One flagella, one end, one mono is Vibrio, and pseudomonas. Then amphitrichous is at both ends. Lopho is tuft or bunch. Bunch is present in Bacters. And peritrichous is going to be present in Proteus, E. coli, and Listeria. But what are the types of motility that you have? The types of motility are in front of you. Firstly, I want to start from this one, that is differential motility. Differential motility is a question that was asked in the INI-CET 2023 exam that was conducted in the month of May. And differential motility tells you that, okay, there is one organism called Listeria and there is one organism coming under the family of Yersiniosis, and they tend to show you a differential motility. Means that they will show you a motility at a low temperature of 22 to 25 degree, they are going to be motile. However, if you put them at a higher temperature of 37 degrees centigrade, they are going to be non-motile. How do I learn it? Because now I will tell you that out of these, Listeria is going to have a tumbling motility. Do you know what is tumbling? Tumbling is like a drunk man who is falling left, right, tully man, right? So Listeria is that drunk, tully, tumbling organism. And if you just go by basic human physiology, it is easier to be drunk at a younger age, but it is very difficult to be drunk and very, very unhealthy for the body, and your liver and your body will not be able to take it at an older age. So it's easier for you to party all night and drink at the age of 22 to 24, where you will be tumbling and drunk and all of that. But at an older age of 37, you will not be able to tumble and get drunk. That is how I have learned it. So Listeria is tumbling at a young age of 22, but not tumbling at 37. This was the exam question that came in the recent exams. Now coming to darting motility. Darting motility is basically a very, very fancy name for something known as a shooting star motility. And let me tell you, there are two organisms in which you will be seeing this. One is going to be Vibrio and the other is going to be Campylobacter. Campylobacter and Vibrio are going to show you darting or shooting star. Next, who shows you corkscrew motility? Corkscrew is reminding me of tight screw. Who is looking like a tight screw? Treponema pallidum is going to be looking like a tight screw. So remember it. Who shows you twitching? T for T, Trichomonas vaginalis shows you twitching. For the ionized aspirants, Ichinella. Ichinella is also going to show you the same twitching motility. Coming to falling leaf, Girta hua leaf. Girta hua leaf is Giardia lamblia. Girta hua leaf is Giardia lamblia. And now coming to what is swarming. And actually, guys, the picture that has been shown over here is a picture of a swarming motility. Something that is going round and round like this. That is concentric circles. That is swarming motility. Where do you see swarming motility? There is a very famous mnemonic that I have taught you, and that mnemonic goes by the name of PVCs. Guys, what is PVCs? Let's quickly make a quick overview of it. Number one, swarming is very famous for Proteus. Then VP. What is VP? Vibrio parahemolyticus. In fact, one more Vibrio can show you this, and that is going to be Vibrio alginolyticus. So the rare wala Vibrios show you. C. One Clostridium, one Bacillus that starts with a C. So Clostridium konsa? Clostridium tetni. And which Bacillus starts with a C? That is Bacillus cereus. Finally, we have S for Serratia. These are all the organisms which are very famous for showing you the very, very sought after question, that concentric circles wala swarming. You learn it by the mnemonic PVCs. P for Proteus, VP for Vibrio parahemolyticus and alginolyticus, P for Clostridium tetni and Bacillus cereus, and S for Serratia. These are the alphabets of PVCs that you have to remember, and very, very important question for the exam. Yes, this is what I'll write. I'll write PVCs in front of it. Correct? Now you will ask me that this was about the flagella. What is it going to be about, the fimbriae or the pili? So please note, fimbriae or pili are the ones that were very tiny and they're going to help in adhesion. So for example, in a while I will be teaching you bacterial genetics, in which I will be teaching you something known as conjugation. So where are fimbriae or pili used? Whenever one bacteria and another bacteria want to connect. So this white color pencil of mine is the pili. Whenever they want to connect, they're going to have a pilus which will help in connecting. This is a question which again I would want INI-CET aspirants only to learn. Not important for NEET and FMGs. How do you test for fimbriae or pili? There's a test which is referred to as heme agglutination, and there is a test which is referred to as surface pellicle testing. So now a question that I want to ask all of you is, all practical purposes I am saying fimbriae or pili are for attachment, but can they help in motility? Can they aid in motility? Yes, this has also been a recent exam question. And first organism in which you need to know where they help in motility is Neisseria, and the other one can also be Pseudomonas. And let me tell you, the kind of pili which helps in this motility is going to be the type 4 pili. So when you go to the individual topics and study, you will realize in Neisseria, I've told you it has a type 4 pili. Neisseria is a focus. You don't expect it to be motile. Neither will I teach you under any kind of motility. But can it be motile? Yes. Why? Because it has a different kind of a pilus, not a flagella. It has flagella nahi hai. It has a pilus which is helping it. Pseudomonas can have a flagella also. Yes, remember I told you pseudomonas can have a flagella, monotrichous flagella, yes, but it also has a type 4 pilus. So this is something extra again for the Central Institute exams that I'm telling you. And on that note, if you go back to your diagram, you will realize that you have wrapped up all of your bacterial anatomy. Done with bacterial anatomy, let's move forward to a very short topic, and that is going to be the growth curve, means bacterial physiology. Now we all know that these are the different factors that a bacteria would need for its growth. For example, it will need oxygen. If it needs oxygen, you will call it an aerobic organism, aerobic, and if it does not need oxygen, you will call it an anaerobic organism. So if you ask me what are the obligate aerobes and the obligate anaerobes. So first and foremost, obligate aerobes means aerobes is something that requires oxygen. Let it breathe. Breathing se related kuch hoga. So must let nagging pets breathe. What is must? So as you can see in front of you, the must M is going to stand for the MTB. L is going to stand, or LE is going to stand together for Legionella. Then we have nagging. Nagging will stand for nocardia. The pets will stand for Pseudomonas, and breathe. Breathe has the maximum B's associated with it. It has three things, that is Bacillus, Bordetella, and Brucella. So must let nagging pets breathe. Repeating: Mycobacterium, Legionella, nocardia, Pseudomonas, Bacillus, Bordetella, Brucella. These are all obligate aerobes. Which are the ones which do not require oxygen? The ones which are choked by air. They do not require oxygen. So you could have learned it as ABC also, but then it's difficult to learn such mnemonics. Now at least you know that, okay, the ones which don't need oxygen, if you give them, they will be choked. They are choked by air, or ABC. What is choked by air? So Clostridium, Bacteroids, and Actinomyces. Clostridium, Bacteroids, and Actinomyces, they are going to be obligate aerobes. And that is what I realized, that every family, if I pick up, one member of the family needs oxygen, one member of the family does not need oxygen. And I'll give you an example. So for example, you pick up the spore-forming organisms. One of the spore-forming organisms is Bacillus, and the other one is Clostridium. And I realized that Clostridium has come over here. It does not need oxygen, whereas Bacillus had come in the previous one, means it needs oxygen. Similarly, if I pick up the filamentous organisms, what are the filamentous organisms that we have? Number one, we have nocardia, and the other one we have actinomyces, right? So can you see actinomyces is coming up in this list above you, that it does not need oxygen, and nocardia was coming in the list before this, that it definitely needs oxygen. So if you pick up a family, one of them needs oxygen, one of them does not. Repeating: must let nagging pets breathe are obligate aerobes, and choked by air are going to be obligate anaerobes. In fact, you also need to know what is the list of the facultative anaerobes, and I've not got you any kind of mnemonic for it, because now whatever is left comes under facultative anaerobes. For example, staph, strepto, enterobacteriaceae, hemophilus, vibrio. If you go chapter 1, chapter 2, chapter 3 in bacteriology, everything comes under here. The best is that learn obligate aerobes and anaerobes. Everything else fits in over here, but one tiny little exception, which are the microaerophiles. Little amount of oxygen they will survive in. What is that little amount of oxygen? 5% oxygen they require, and these are the bacters. Bacters matlab I'm talking about the Helicobacter and the Campylobacter. So these are the bacters that we are referring to. So coming all the way back, I've spoken about the need for oxygen, but some of them say that we need carbon dioxide. Carbon dioxide is capnophilic, and one example of a capnophilic organism is going to be Campylobacter. The usual pH that they require is going to be 7.2 to 7.4 or 7.6 also, but I think all of you know that there is one organism that is Helicobacter pylori, and Helicobacter pylori is definitely something that can survive in an acidic pH. Similarly, all of you know there is an organism called Proteus. Proteus tends to survive more in an alkaline pH. These are all clinical things that you have studied now. That is why Helicobacter pylori tends to survive more in the stomach, because the stomach has an acidic pH. Proteus causes alkaline urine, alkaline environment where it is going to survive. So this is what I know. Similarly, some of them will need light, some of them will not. But the most important question that you get is on temperature. See, the ones that survive in a medium temperature of say 20 to 40 degrees, or in the books it will be written as 25 to 40 degrees, somewhere in a medium range, they are mesophiles. The ones that require a very high temperature of 55 to 80 degrees. And I'll give you an example for this. For example, you must have heard of Bacillus geothermophilus. Geothermophilus. The word only tells you that thermal is coming. It needs a high temperature. And the ones that are psychic matlab they need low temperature. That is less than 20 degrees, even sometimes into minus. Those are psychrophiles. So remember, mesophiles are medium temperature. Thermo or thermal means high temperature, and psychro or psychic, they require low temperature. These are the different needs that a bacteria has. But how does a bacteria grow? And that you need to study with the help of a bacterial growth curve. You can all see that there are different phases of a bacterial growth curve that are present over here. So what if I tell you the x and the y axis? On the x axis over here you have the time, and the y axis has the number. Means that, okay ma'am, from one bacteria it is dividing into 2, from 2 it is dividing into 4, from 4 to 8, 8 to 16, so on and so forth. That is the bacterial growth curve. But in the first stage I realized nothing is dividing. The number is the same. There is a lag. The next one is going to be log. It's a very common sense thing. I can see the number is rising. So matlab exponential hoga, log hoga, but still sometimes common sense leaves us in the exam, exits the room. You enter the room and your common sense leaves the room and you enter the exam hall. So still for that case, keep it in alphabetical order. Lag comes first, log comes later. After that comes a phase where everything becomes stationary. So that is the stationary phase. And then it starts dying. So that is known as the death phase. So do you want to do the features of the lag and the log and the stationary and the death phase? So remember, lag is like a lockdown. Means only the cell size is going to increase. That's it. Number is the same. Number is not increasing. They're not dividing. Basically, why did I say lockdown? During the COVID lockdown, what did you do? You just sat at one place, did no work, sat at one place and became fat. So if I ask you, what is the maximum size? Maximum size is obtained at the end of which phase? So maximum size is obtained at the end of the lag phase. Ma'am, lockdown ke end par, yahan par, we were the fattest. After that, lockdown opened, we had to work, we had to divide, we had to earn for ourselves, work for ourselves. So we started dividing. So if they ask you, when does cell division occur? Cell division occurs finally in the log phase. If you start working, will you lose weight? Yes. So can I say the cells become smaller in size? Because if someone is working, someone is dividing from 1 to 2, from 2 to 4. If someone is dividing, means the person has started losing weight. cells become smaller. If they ask you this is going to be maximum metabolic activity, of course this is going to be maximum metabolic activity because it's dividing. So these are the features of the lag phase, of the log phase. Log means that finally it is dividing. Comes to a phase where everything becomes stagnant. Now the bacteria says all my nutrients have been depleted. I cannot divide any longer. Now I have reached a plateau. Like you're studying too much. Then come the phase of saturation. That bus ma'am, I cannot study any further. That is known as the stationary phase. What are the things that you have in stationary phase is so easy to learn, because if you remember stat, from stat for stationary, you will remember stab, and means what? The spore formation will occur here, toxin production will occur here, antibiotic production will occur here, bacteriocin production will occur here. Repeating, sporulation, toxin production, antibiotic production, bacteriocin production. The stab formation will occur in the stat phase or the stationary phase. And after that, of course, the bacterial count will go down. Why? Because the bacteria has started dying. Either cell death has occurred or involution has occurred. That is why the count is going down. So let me ask you quick questions out of this. If I say the cell is maximum in size at the end of which phase? At the end of lag phase. Where is the cell dividing? It is log phase, maximum metabolic activity. Where is it going to have stat stab? Matlab, where is it going to have sporulation, toxin, antibiotic and bacteriocin production? In the stat phase. Finally, when is it going to die off? In the death phase. That is the entire bacterial growth curve, and you get a lot of questions, especially this sporulation wala PYQ is very, very famous in the previous year papers. And yes, that wraps up all of the bacterial physiology also. Hi everyone, let's begin with the next topic, and that is the bacterial genetics. This also comes under the general microbiology and is very, very important, not only because this is a topic which has come a couple of times in the previous year papers, but also important because it's a little complex. It's got a couple of formulae or kind of things which are not easily remembered in one go, and that is why the examiners like asking questions on these. So let's get going. What exactly would I be teaching you in bacterial genetics is how the genes are transferred, which means that from one bacteria to the other, if I have to transfer something, how am I going to do that? And that is the entire concept of this particular topic. So there are three methods that you have. The first one that I'm starting with is transformation. The second one that I'll pick up will be transduction, and the third one will be conjugation. Okay, let's get going. The first one, what did I say? The first one that we'll study is transformation. What is transformation? I'll explain that with the help of a diagram. So for example, if this is a bacteria, this bacteria, and like every bacteria I have taught you, doesn't it have its own DNA? Every bacteria has a circular DNA that is available, right? Now if this bacteria says I want to pick up some DNA from some other bacteria, what will it do? It will just pick up some random free DNA from the environment. Maybe some bacteria had died and that bacteria's DNA had been released, so random free DNA from the environment will be picked up, and that is known as transformation. So please remember, it is the transfer of the free DNA that is going to occur. So where does it occur through? How is the DNA coming in? Did you see? How is it coming in? It is coming through the cell wall. That is question number one. Question number two, when does this happen? Before this, did you guys all go through the growth curve? So which phase of the growth curve does this happen? This happens during the log phase of the growth curve. And finally, is there any material or factor that a bacteria requires that it can undergo this? Yes, what is required is the competence factor. What is required is going to be the competence factor. So repeating once again, what we need is that it has to come through the cell wall, and that will happen during the log phase, and it will require the competence factor. That is what we call as transformation. One of the examples of a bacteria that tends to undergo this is Streptococcus pneumoniae, on which previous year questions have also been asked. So that is how the bacteria, see, now something new will come inside it, new properties will come, and that is the concept of transformation, which is done. Now I move on to the next one, that is transduction. What is transduction? For transduction, remember, this is the most common mode of inheritance that you have in Staphylococcus aureus. I'm going to make a small table at the end at which bacteria is having resistance via what, but for the time being, please note, for Staph aureus, the most common mode of getting drug resistance is trans. You must have heard that, you must have read in bacteriology segment that for Staphylococcus aureus there is penicillin resistance, beta-lactam resistance. How does that resistance happen? That is happening by most commonly transduction. So what is transduction? This is going to be done by, I'm going to draw a diagram. Let's see if you can identify what exactly is this. This is a bacteriophage. So exactly what you want to know next. What is a bacteriophage? Bacteriophage sounds like a bacteria, but it is not a bacteria. Bacteriophage is actually a DNA virus. So if someone asks you, is it a virus? Yes. And what kind of genome does it have? DNA or RNA? It is a DNA virus. And what is this shape that I've shown you over here? It is a tadpole shaped structure that we have. So let me show you now the different types of transductions that are possible. There are two transductions that you have to know in literature. One is known as a generalized transduction, and the other is known as a specialized transduction. So let's get down to describing it. So in the textbooks now, it's very, very complicated. I like to give you a day-to-day example which all of us are guilty of, and we have gone through it. We practice it every day, and that is ordering food via some or the other online platform, right? We do that. So what is the purpose of an online platform? What do the food delivery apps do for us? They pick up the food from the restaurant and they come and give the food to us. So it's a delivery service. Same way, I would say bacteriophage is like your food delivery app. It is going to pick up a DNA from one restaurant and is going to come and give the DNA to you. So there is, the restaurant is the donor, you are the recipient. So pick up the DNA from the donor, give it to the recipient, and that is what it has. So now let me explain to you the first one, that is what is generalized transduction. Look at the labelings very carefully. This what we have is the donor bacteria. This is the restaurant from where I want the food should be picked up, and that food should be given to the person who's waiting for the food. So that is going to be recipient bacteria. Who is going to pick up the food from one donor DNA and give it to the recipient DNA? There comes the very famous food delivery app. So this is a bacteriophage. So this is the delivery partner. This is your delivery boy who has reached the restaurant. After reaching the restaurant, it first enters the restaurant. And you see the bacteriophage has given its DNA. So now you say that, ma'am, there are two people standing inside the restaurant. This is the owner of the restaurant. In other words, what is this? This is the DNA of the donor bacteria. Now the Swiggy, Zomato, the online app person has also gone inside. So can I say this is the bacteriophage DNA that is inside the restaurant, and the donor DNA is also going to be inside the restaurant. So all in all, I realize there are two DNAs who are sitting inside it. What happens? We say, okay, let us make new and new. Let us make more and more and more of DNAs that are there. So you can see many of these DNAs that have been made over here. But what happens? I don't want these DNAs. I want these DNAs to be transferred. So now new bacteriophages will form. New delivery boys are coming. So did you realize some of the bacteriophages had their own DNA, whereas some of the bacteriophages have the donor's DNA. And that bacteriophage you are interested in. You are interested in the bacteriophages which are carrying the donor's DNA, because you want that the donor's food item should be delivered further. And can you see these have gone and have given the food item onto the recipient? Can we all see that? They have gone and given the food item to the recipient, and that is what has happened. Finally, the donor DNA has successfully been transferred to the recipient via what? Via the online food delivery app, and that is bacteriophage. That is specialized. Apna DNA kept. Whereas that is generalized. Whereas if I tell you what is specialized or restricted transduction, something special is going to happen over there. What is special? Same, see, story is still the same. Again, you have a donor bacteria that is present. After that, again, you have a recipient bacteria that is going to be present. Again, you are utilizing the same food delivery app, that is the bacteriophage. The only difference, now let's see what all is common. Ma'am, same, the food delivery person came, he gave his own DNA, the donor had the other DNA. The problem that I see over here is that the food delivery DNA, that is bacteriophage DNA, and the donor DNA did not keep separate. They decided, okay, let us get into a relationship. Let us first get into, they've become best friends, they've gotten into a relationship. So basically there is a fusion that has happened of the bacteriophage and the donor DNA that is available. Okay, but after every relationship, there is going to be a breakup also. After some time, we realize that both of them end up separating out, right? So first the bacteriophage and the donor DNA decided to get into a relationship. After some time, they ended up having a breakup. That breakup over here is known as induction. What is induction? Induction means that the bacteriophage and donor DNA separate. But tell me, whenever a breakup happens, breakup is never going to be okay, apna apna. You take some gifts from one person and the other person takes away. Everyone takes some gifts and some money from here and there, some memories if you don't want to be materialistic. So when this bacteriophage DNA got separated, we realized that this bacteriophage DNA also ended up taking few memories from the donor. Can you see? It's a combination form, because breakup is not without memories and without gifts. So breakup hua, but obviously a little bit of memories have come. Now can you imagine the new bacteriophages that will form will not have the original only donor DNA. They will have a combination. They will have a combo that will be present. So what is going to get delivered? What food item is going to get delivered to the recipient, ma'am? The combination food item, the combo DNA is going to get delivered to the recipient. And that is the difference that you have between generalized and specialized. If you remember, in generalized, it was a simple food delivery. The bacteriophage went, picked up the DNA from the donor, went and gave that DNA to the recipient. Simple. Pick up the food from here and give it to the recipient. But in specialized, there was a complication. Complication kya hai? Relationships. First there was a relationship, then there was a breakup, and that combo DNA was ultimately getting delivered. And that is what you call as specialized DNA or specialized transduction. So my rule in the exam is, how do I know that the examiner is talking about transduction? I have to find for this bacteriophage. Agar bacteriophage dikh raha hai or bacteriophage dikh raha hai, matlab the stories of transduction. Then I just have to find out generalized hai, specialized hai. Generalized, keep it very general, very simple. Pick up the DNA from here and give it to here is general. Specialized is that complicated story of fusion and then separation and all of that. But now I want to ask you that, these, I have to tell you a little bit about the food delivery boys also. You have to give them importance. So what is it about the bacteriophage cycle that you have to know? So talking about the bacteriophage cycle, please note that this bacteriophage cycle is going to follow number one, a lytic cycle. Lytic means what? Actually, whatever I was telling you till now was lytic only. You did not realize. If I show you the next cycle, did you see that, okay, so many food delivery boys were there, but how are they finally coming out? By breaking the bacteria. How are so many food delivery boys have been made over here, but how are they finally going to come out? They are going to break the bacteria and come out. So that kind of a lysis or cycle is known as a lytic cycle. Whereas what was that cycle in which I told you breakup hua, patch up hua, fusion happened? So this fusion wala scenario is what I want to describe with you, because that is known as lysogenic conversion. Now this is a question that you got in your previous year papers. What is lysogenic conversion? Lysogenic conversion means that जो bacteriophage का DNA है and donor DNA. What did I say? The bacteriophage DNA, the food delivery boy's DNA, and the donor, that is the recipient DNA, they ended up undergoing integration. And after that, was a proper relationship ban gaya na, but after that, a breakup happened. It was such a strong relationship that it started getting transferred onto the kids. It started getting vertically transferred to the offspring. So that is known as a lysogenic conversion. There is a transfer to the daughter cells that I am seeing over here. Has everyone understood what is lysogenic conversion? We don't have to be so negative na, कि if relationship has happened, then breakup will definitely happen. That is not always the case. 1% are successful love stories also. That if a relationship has happened, if the bacteriophage DNA and the donor DNA have fused, maybe this is a successful relationship, and maybe this will be transferred onto the further progeny or the children, and that is known as lysogenic conversion. Why am I teaching you this story? Because I had to end at a positive note. Else you would have told me, ma'am has taught us that every relationship has a breakup. No, I'm telling you that some relationships are successful also. That is lysogenic conversion. But why am I teaching you this? Because of previous year questions are that which are the toxins that you have in the bacteria that are going to follow this lysogenic conversion story, and that is A, B, C, D, E. So how I have learned it is lysogenic conversion is basically related to transfer to the daughter cells or the offsprings. And offspring offspring सबसे पहले क्या सीखेगा? What will the offspring learn? The tiny little kid baby will learn A B C D E. So A B C D E is what is following the lysogenic conversion. What is A B C D E? The A and C toxins of Streptococcus pyogenes. So A and C toxins of Streptococcus pyogenes. Then B say the B and D toxins of botulinum toxin. So B for botulinum toxin. A is going to be A and C of Streptococcus pyogenes. Then C क्या है? Simple. C for I only know cholera. So cholera toxin. D I know which bacteria, diphtheria toxin. And E for enterohemorrhagic E. coli. If you read the bacteriology section, you know that enterohemorrhagic E. coli has what kind of toxin? If I had to put up, enterohemorrhagic E. coli has the Shiga-like toxin or verocytotoxin. So what is A, B, C, D, E? Repeating once again for all of you. A is the A and C of pyogenes. D is botulinum. C and D of botulinum. C is going to be cholera. D is diphtheria. And E is EHEC. So remember the conversion that is going on to the children. Children will learn A, B, C, D, E, and that wraps up all of your transduction also. So transformation is done. Transduction is done. Now the last one that we have is going to be conjugation. So let me just find out and ask you something over here. What do you think is this? Is this transformation or transduction? You'll say, ma'am, I can see a food delivery service. I can see a bacteriophage over here. If I can see a bacteriophage over here, the story has to be a transduction. Common sense. What is this? इसमें तो बस uptake हो रहा है. Some random DNA is being picked up. If some random DNA is being picked up, that is known as transformation. What is the last one? The last one is referred to as conjugation. What do you see in conjugation? Some contact is forming. If this is a one bacteria and this is another bacteria, you will see there is a bridge between the two. The bridge dikhega, that is known as conjugation. So let's start studying conjugation from now onwards. So remember, conjugation is by a conjugation tube, and that conjugation tube is made by pili. What is pili? मतलब ये एक बैक्टीरिया है, ये एक बैक्टीरिया है. A tube will form. That is known as pili. Do you remember what was pili? I had taught you in your bacterial anatomy. Pili was nothing but fimbriae. Exactly. So ultimately, it is the fimbria that helps in the transfer, and that is known as conjugation. So do you know? Overall, in the entire bacteriology, the most common mode of resistance that you have is going to be via conjugation. In the entire bacteriology, anywhere they ask you, "Achha, this antibiotic resistance is due to transferred from one bacteria to the other. How you use conjugation?" But in Staph aureus, remember, transduction is most common. The most common, even more common than conjugation, is going to be transduction. And for Streptococcus pneumoniae, I had told you that it is going to be transformation. So this is your final table, and please guys, I am requesting you, don't send me screenshots later on saying that ma'am yahan alag likha hai, wahan alag likha hai, because you will get different controversial answers. I'm giving you the best possible answer from the most standard textbooks that you have to follow. So don't unnecessarily get confused. Remember, the most common mode of resistance is conjugation. However, in Staph aureus it is going to be transduction common, and in Streptococcus pneumoniae transformation is more common. So do you want to finally learn a little more about conjugation? Let's get going. So I've got you something over here. I want you to label it. You guys have that knowledge. Ab bas little testing of your knowledge I'm doing. What do you think are these layers? You say, ma'am, these are kindergarten questions. Capsule hai, cell wall hai, ye sab pata hai. Tell us something new. What do you think is this? You say, ye bhi pata hai, ma'am, every bacteria has a DNA. Again, tell us something new. Now I'll tell you something new. Other than the DNA, some bacteria also have another DNA called plasmid DNA. So a lot of people ask me, what is plasmid? Main DNA nahi hai. Main DNA to every bacteria has to have. So every bacteria has to have a DNA, but plasmid DNA may or may not be present. So what exactly is plasmid DNA? It's again round. I could see it is double-stranded because every DNA is double-stranded, but it is different from the usual DNA. See, it's not a part of the chromosome wala DNA. It's separate. It is going to be extra chromosomal material that you have. So extra chromosomal circular DNA is referred to as the plasmid. But if you feel that, oh, this plasmid is only carrying DNA, no, there are so many things. So if I draw this any one plasmid, for example, I take it up and I draw it over here. For example, if this is a plasmid, you know, it can carry, it's like a taxi, it's like a cab. It can carry any passenger. If it carries passenger F, you will say, oh, it's carrying the fertility factor. It can carry the fertility factor, means now I will call it the F plasmid. Then you say, no, ma'am, the next passenger, not fertility factor. It is carrying something called R, means it is carrying something for resistance. Maybe if this goes, then I call it R plasmid. That means that if this plasmid goes to a bacteria, it will become antibiotic resistant. So on that basis, there are different kind of plasmids that have. This plasmid can be a fertility plasmid. This can be a resistance plasmid. Yahan pe F plasmid likha tha, so what plasmid do you think this is? This is a fertility plasmid, and that is what I want to teach all of you when it comes to conjugation, fertility. So let me give you an example. Ye dekho. What is this? You say, ma'am, this is the bacteria, but do you want to call it a male bacteria? This is also a bacteria, this is also a bacteria. Which is male, which is female? How will I get to know? You will say, dekho ma'am, DNA to everyone has to have. So DNA to hai. But I realize that this one has the fertility factor. Anything that has the fertility factor becomes the male bacteria. This one does not have the fertility factor, so this becomes the female bacteria. How will this male end up giving the fertility factor to the female? Kya banega beech mein? We know that you've told us there will be pili or a conjugation tube that will form. Fine, the tube has formed. Contact has occurred, the tube has formed. But what after that? It is, see, many students believe it is cut and paste. No, remember in Windows we do cut and paste. I'm not doing cut and paste. I'm doing copy paste. What do I mean by copy paste? One of the copies of the plasmid कॉपी करो एंड यहाँ पे पेस्ट कर दो. डोंट कट एंड पेस्ट. डोंट फील दैट प्लाज्मिड इज गोइंग फ्रॉम हियर टू हियर. वो मूव करा एंड वहां चला गया. नो, इट्स नॉट दैट. इट इज कॉपी पेस्ट, which means that one of the copies is going to go over here. So this was always F plus, this is still F plus. Can I say this was F minus and has converted into F plus? So indirectly, if I have to make a table over here, can we say that initially the male was F plus and the female was F minus? But in the end, what happened? Both of them ended up becoming F plus, F plus. I hope that is making sense. It's a copy paste. It's not a cut paste. Remember that. So both of them end up becoming F plus, F plus. That is the question that you get. When a male and a female will conjugate, both of them will end up becoming fertility factor positive. Do you think such a simple question will come? No. Now let me tell you the other two scenarios which are more commonly asked in the paper. Now, first, don't get confused. Same donor bacteria hai, ma'am. Let me see, is it a male or a female? Okay, this must be the usual wala DNA. And this is looking like the fertility factor. So fertility factor to hai, ma'am, this means it is a male DNA. Now, should it go and give it to the female DNA? Of course, but complication of a relationship coming up again. These two decide that, array, what will we go to the female? Let us have a relationship with each other only. Both of them decide that we are going to fuse. Can you see that the DNA here and the fertility factor here have basically fused? This is known as HFR. HFR means high frequency recombinant. HFR basically means something has recombined. It is high frequency recombinant that has occurred over here. This is the relationship. Now they say, okay, we are a couple. High frequency recombinant, we are a couple. Now they say, oh, should we try and go to a female? So can you help me draw a female bacteria? How will you draw a female bacteria? You will say, ma'am, DNA to chalo, everyone has, make a DNA, but don't make the fertility factor. Let her be fertility negative. That is how she will be a female, right? This HFR says, okay, now both of us are in a relationship. Let us go and get into a female bacteria. But possible hai, tell me, when someone is in a relationship, you never travel solo now. Will this fertility factor, this red wala fertility factor, will it be able to transfer alone? No, ma'am, no solo trips allowed. Now they're in a relationship, they will travel together. Same way going for the donor. So now you will say, they will say, acha, theek hai, we can't travel solo, so we will travel saath. So little bit of the donor DNA goes, little bit of the fertility factor goes. What did I say? That a part of the donor DNA is going to get transferred and a part of the fertility factor ends up getting transferred. You say, maza nahi aaya. What kind of a trip is this? Little bit of donor has gone, little bit of fertility has gone, pura nahi gaya. So this female will not be happy. Female will say, what is this? You've given me compensatory gift. Full fertility factor has not come. Little amount of fertility factor has come. So I'm not going to become positive. I will still remain negative. If this female has, she has taken offense that what is this, that little pieces are coming to me. I'm not some time pass story happening here. So she's not happy. As a result, in the end, she said, I will still remain F negative only. I'm not happy. But don't you feel you created a full mess? She had her own DNA. Then you got after her own DNA, you got a little bit of donor DNA. A little bit of fertility factor, thoda, you have created a full recombinant mess over here, which means that I can finally tell you that when HFR is going to combine with a female, no one is going to get anything out of it. The female will still remain the female only. She has taken offense that this little things are coming to me, I'm not happy, but she has become a recombinant female. But remember, female remains female because relationships are complicated. You have a relationship here, the story is going to be complicated only. She does not get converted into a male. Relationship story ho gaya. So what do you think the last story is going to be? Breakup. Everyone, third scenario, please start labeling with me, then we'll make a summary. What is that? Okay, this must be the donor bacteria. Can you tell me whether it's a male or female, ma'am? Fertility plasmid hai. If fertility is there, it is of course going to be a male over here. Then you'll say, ma'am, in the next one, I can see aapne to fuse kar diya. The male has basically undergone fusion. The male has made HFR. But in the next story, you have decided that the fusion has ended up in a breakup. Breakup, can you see? The fertility factor has gone separate and the DNA has gone separate. But what did I teach you earlier also? Breakups are never without memories. So this fertility factor has taken a part of the donor DNA also while breaking up. Now what will you call this? You will call this F prime. What is F prime? F prime means that this is going to be the same fertility factor, but it has got a little bit of memories of the donor with it, and now we call it F prime. Now imagine if this F prime goes to a female. Female matlab, ma'am, fertility factor nahi hai, that is a female. Imagine if F prime, copy paste kar do. So if F prime goes to a female, the female will also become F prime. So this time the female is going to convert into what? This time the female will convert to F prime. So that means if the fertility male is an F prime and he combines with the female, both of them end up becoming F prime, F prime. So how can I learn all of this in an easier manner? देखो, जो भी male होगा ना starting में, whether the male is F plus or the male is F prime, if that is going to combine with the female, female says अच्छा, जो भी आ रहा है, I will accommodate. If F plus is coming, female says I will become F plus. If F prime is coming, female says I will become F prime. So female is very adjusting. Whatever the male is ready to give, the female is ready to take. But वो relationship वाला story complicated है ना. When HFR is being proposed to the female, now the female is not happy. Now the female is just going to remain a female, but a recombinant female. The easiest way of learning is, if the scenario is simple, the female says, okay, I will adjust, I will become. So F plus hoga, so I will become F plus. F prime hoga, I will become F prime. But if this HFR story is being presented to me, then I shall remain the female only, that is a recombinant female negative. That is the summary of this entire story. But why was I focusing only on fertility? Can the plasmid also carry something else? Yes. Fertility story done. The plasmid can also carry the resistance factors. That means that, for example, this was a plasmid. Can you see resistance to so many drugs are being carried at the same time? So this entire plasmid has one big loop. This big loop is known as RTF, resistance transfer factor. So this big R plasmid is known as RTF. On that, many drugs can be carried at the same time. These small R's are known as resistance determinants. So look at this. This one RTF is carrying resistance to so many antibiotics at the same time. This is going to be so dangerous. Now imagine if this one ends up going to a bacteria, in one go the bacteria will become resistant to 10 antibiotics at the same time. So this is going to be very dangerous and very difficult to treat. And that is what is known as transferable drug resistance. Now the next topic that I'm teaching you related to this is only for NEET and INICT students, not for FMG students. So please remember, they ask you, what is the difference between transferable drug resistance and mutational drug resistance? Transferable drug resistance, I've understood, is because of the resistance transfer factor and is because of all those R determinants and conjugation and all of that. So ma'am, this is very difficult to treat and it is going to be major problem. There is mutation. Mutation to single drug ka hota hai. Like for example, TB mein, in your medicine lecture you study, TB mein there's a very common mutation known as rifampicin drug mutation or rifampicin drug resistance. Why does rifampicin drug resistance happen? Because there is a mutation in the RPOB gene, right? The RPOB gene has undergone a mutation, so single mutation hua to single drug ka problem hai. So this is obviously easier to treat because this is going to result in a single drug resistance and this is going to be easier to treat. So this is a question that they ask you in the paper. Transferable drug resistance will transfer too many resistances at the same time. So it will be very difficult to decide now what antibiotic should I use. Whereas mutational drug resistance is a resistance to a single drug. So you can easily change the drug, use an alternative form and very easily treat the situation. All said and done, this becomes your final story that you had to know, guys, for these three types of resistance transfers, and that is transformation, transduction and conjugation. Before I finish this bacterial genetics topic, I just want to touch upon something, and that is known as CRISPR-Cas9, because I know you guys have been struggling a lot with this, be it path, micro, biochem. This one genetics topic I want to quickly pick up because while you study this for humans, I want to tell you CRISPR-Cas9 technology actually started in bacteria. So first I want you to know CRISPR क्या है and Cas9 क्या है. Cas9 is said to be, in simple word, Cas9 is a scissors. So I will tell you wherever the scissors are needed, कुछ cut करना होगा, I will use Cas9. What is CRISPR? So now actually CRISPR is a short form that they have made out of this word. What is CRISPR? CRISPR means there is going to be clustered regularly. So write with me and also note how we'll make a diagram. Clustered regularly. Something is going to be clustered, okay? Interspaced with space in between. So there is going to be interspaced short palindromic repeats. Now what do I mean by this short palindromic repeats? Before I start, do you guys know what is palindrome? Just in basic English, what is palindrome? Like for example, Malayalam is a palindrome, which means you read it from left to right or you read it from right to left, so same lagega. Malayalam is going to mean the same. You read mom, left to right to left, same. Dad, left to right to left, same. So everything that you study is going to be left to right to left, same hoga, wo palindrome hota. That is the meaning of palindrome. So now you will realize that you are getting these clusters. Can you see these yellow color clusters that I have made? Yes, okay. And there are spaces in between. Now you will ask me, acha ma'am, what are these spaces? What are these spaces? I call these spaces as screenshots. Remember, spaces are screenshots. Screenshots of what? Let me explain. So ye dekho, this is a bacteria, and bacteria is, if you felt COVID troubled you, viruses trouble you, viruses only trouble human beings, you are mistaken. Viruses are troubling bacteria also. Imagine if a virus is going to cause infection in a bacteria. Viruses are damn deadly. They are not even sparing the bacteria. So if a virus, this is virus number 1. If this virus number 1 came and the virus number 1 had entered a bacteria to apna defense karega. Now what will the bacteria say? Hang on, this green color is a virus. I will kill it. If it wants to kill it, how will it cut it? Tell me guys, kuch bhi kill karna hai, cut karna hai. Ma'am, cutting ke liye you have told us that later on you will be using a scissors called Cas9. So how I have to cut the virus, that I know. But then I also have to remember that achha, this virus had come and created a problem. This bacteria takes a screenshot of the virus. देखो बैक्टीरिया ने क्या करा, स्क्रीनशॉट लिया और सेव कर लिया. That next time when you come, you wait and watch. I will not let you infect me. Same way, this virus came. This virus has, you've taken a screenshot. You've killed the virus. Story khatam. Next time, this orange color virus came. Again, kill the virus. Take a, so it's like a deadly. You see those murder mystery. Those serial killers and murderers, where they kill a person, they take a photo of that person, and they move away from the crime scene. So same way, you kill the virus, you take a photo of the virus, and you keep it in your memory. Next time a virus came, again, kill kara, photo liya, save kar liya. Next time a virus came, kill kara, photo liya, save kar liya. So that is what is being done. You realized all these spacers are memories of the virus, so that next time when the virus comes, the virus cannot affect this bacteria. So can I say CRISPR-Cas9 is a bacterial defense? Like we have immune system, bacteria also has an immune system against the viruses. This is the question that you will get. CRISPR-Cas9 is the bacterial defense against viruses, and that is the story that you had to know. Please remember, in the last point that I want to tell you, because everything around COVID is very important. If you remember, there were 2 scientists from West Bengal who made a test in COVID known as the Feluda test. The Feluda test for COVID was basically based on which technology? It was based on the CRISPR-Cas9 technology. They utilized this technology, and that is how they made a testing called the Feluda test. Yes, on the basis of that, this was just one miscellaneous topic, path, micro, dono mein important hai, and that wraps up all of your bacterial genetics. Hi everyone, let's begin with the last part of general microbiology, and that is sterilization and disinfection. So how do we sterilize? And this is very important not only for your MCQs but also also for your day-to-day practice and how you would be dealing as a doctor in the hospital and the clinics. So when we talk about sterilization methods, we divide them into two varieties, that is physical as well as chemical. Let me start with the physical methods, which is going to include heating, filtration, and radiation. Let's begin with the heating methods only first. And do you know how many types of heat do we have? We have something known as a dry heat. Just imagine heating something with a candle or a gas burner. That is just simple dry heat. Whereas if you start using something like vapors, like where do you think of day-to-day life? Vapors like boiling of water or a pressure cooker that creates vapors, steam, right? So that becomes moist heat. So what do you think is going to be more effective? Of course, moist heat is going to be more effective because it has those vapors which are also going to act as an added heat. So repeating, there are two types of methods, dry heat and moist heat. And under dry heat, let me tell you, the two most important ones that you have are going to be the hot air oven and the incinerator. These are the two questions that you're going to get in the paper. So you must be wondering, how do they cause sterilization? Sterilization means you should be able to kill a bacteria, kill the organism. So how? What is the mechanism out here? Remember, the code for dry heat mechanism is code. Means number one, how is it going to kill? It is going to cause charring of the molecules. Number two, it is going to cause oxidative damage. D, of course, it is going to denature all the proteins. And lastly, it is going to elevate electrolytes. Now, you know that elevation of electrolytes means that the bacteria are going to be killed. So charring, oxidative damage, denaturation of protein, and elevation of electrolytes are the mechanism of action by which the heat is going to kill. And what are the two things under it? We have hot air oven and incinerator. So let me talk about each one of these. Let us first start our story with the hot air oven, and it looks exactly like an oven that you must have seen in your households for baking and cooking. Same kind of oven is there. Remember, the temperature maintained is 160 degrees centigrade for 2 hours. That is the time and the temperature that you have to learn. 160 degrees centigrade for 2 hours. And what all will you put inside it? I will show you the list that is written in the books. So number one, they say glassware and cotton swabs. So I'll draw one thing over here. This looks like a glass flask or some kind of a test tube. So anything that is to do with glass, and how have you plugged it? How have you stopped it? You have stopped it using cotton. So number one, anything glassware and anything cotton swabs is going to be sterilized in this. Apart from that, how I have learned it is a rhyme. Anything that is going to be dusty, anything that is going to be oily, anything that is greasy, and anything that is liquidy. These are the things that are going to be sterilized inside a hot air oven. So dusty, oily, greasy, liquidy means dusting powders, the powder that you use for dusting the surfaces, the oils, the grease material, and liquid paraffin. So dusty, oily, greasy, liquidy. These are the things that you can sterilize in a hot air oven. But hang on. How do I know that this temperature was definitely maintained and 2 hours have definitely passed? Everything had happened correctly. To know that everything had happened correctly, I use a control. And what is the control that you use in this situation? Either you can use a bacillus species or a clostridium species. Any reason why am I either using bacillus or clostridium? Because these are the spore-forming organisms. I know from my previous lectures that spores are very, very difficult to kill. So if something can kill the spores, if you can kill these, means, but yeah, the method is very, very good. So the point is that you should be able to kill these. You should be able to kill these, and then means your methodology is very good. So what is it? Bacillus subtilis, Bacillus atrophius, they're almost like cousin brothers. And what in Clostridium? Clostridium tetani. This is what you have to know for hot air oven. You say, ma'am, is it necessary to use this as a control? Can I use something else? Yes, please remember there is a chemical method also. This is not important for the FMG exam, but for NEET and INI it is. There is a chemical control that is used, and that is known as the Browning's tube. You should just know the name. And this is a tube which has a red color fluid. So when I keep this tube, for example, if this is a tube, this has a red color fluid, I keep it inside over here along with all my oily, greasy, dusty material. I will keep that tube over here. At the end of the procedure, it should have changed to a green. If it has changed into a green, I am happy that okay, my procedure has happened very, very well. So that is known as the Browning's tube. It has to go from red all the way to green, and now we are sorted. So that is going to be a chemical method of control. Whereas when I'm using the spores, that is going to be a biological, because I'm using biological spores, right? That is a biological control that I've used. So coming back, number one, I have to do hot air oven, which is done. There's another dry heat method, and that is known as incinerator. So I don't know how many of you have noticed, but please do that. All your hospital waste goes to a very, very big building kind of a machine. It's a huge machine in a building, and that's present in every kind of hospital, and that is hospital waste or any kind of human body parts. Where are all these things going? That is going into the incinerator. So that is supposed to have a very, very high temperature so that it can narrow down all that big waste of the hospital into lesser amount. Basically, bulk kam karna hai. You have a lot of bulk generated in the hospital. You have to reduce the bulk. So there are two chambers. There are two chambers that every incinerator plant has. The first chamber has a temperature of roughly 700 to 800 degrees centigrade, and the second one has roughly 1000 to 1100 or 1200 also. So you can learn any one range. So 700, 800 and then 1000 gas pass. These are the two chambers. You have to make the material pass through both. First, you will make it pass through a low temperature of 700, 800 degrees centigrade, and then a high temperature of 1100 centigrade. So that is what you have for incineration, and that wraps up your first methodology, and that is dry heat. But I told you now, what is even better than dry heat is you can create vapors. If you can create vapors and moisture, that is even better. So that is moist heat, and moist heat is something which you are going to divide into three parts. Less than 100 degree if you create vapors, at 100 degree if you create some moist heat, and above 100 degrees centigrade if you create moist heat. So please remember this table. This is the most useful table that you can have for moist heat sterilization. So what did I say? For moist heat, you're going to consider below 100 degree, at 100 degree, and above 100 degree. Very easy to learn because we are all cricket lovers, right? So we have to learn the cricket wala mnemonic, IPL. And how is IPL famous? Batting is very, very important in cricket. That is what creates all the entertainment and excitement, right? So remember, IPL and bat is how you learn this table. What is IPL and bat? IPL and that, L will tell me low. How do I learn what is below 100 degree? I and P. I means there is inspissation and P means there is pasteurization. So repeating once again, inspissation and pasteurization are below 100 degrees centigrade. So what are these? Let me tell you, pasteurization, so I don't think you need to know where is pasteurization done. It is done for milk. There are two methods, holder and flash. Which of them is going to be flash? Jaldi jaldi. Of course, ma'am. Flash is going to be jaldi jaldi. Holder is going to be little longer. So remember 6330. What did I say? 63 degrees centigrade for 30 minutes. I'm holding it for 30 minutes. So I have learned it as 6330. That is Holder's method. Whereas 7220 means 72 degree for 20 seconds. See, I said seconds mein khatam ho gaya. That is why it's flash. And remember, after this, you can't keep the milk at 72 degrees. After 7220, the 20 seconds ke baad, you will cool it to less than 13 degrees. So everyone, what is Holder's? You hold it for a long time. 6330 minutes. Whereas flash method is flash, 7220 seconds. But now, which is better? Remember, flash method is considered better because it can kill something which no one else can kill, and that is Coxiella. The flash method can kill Coxiella, and that is why it's considered better. So that is pasteurization of milk. What else can come under 100 degree? It is inspissation. I have learned inspissation as 80-20, 80-20, 80-20. Matlab means you will say that, ma'am, 80 degrees centigrade for 20 minutes, then again 80 degrees centigrade 20 minutes, then again 80 degrees centigrade 20 minutes. Is this a 3-day procedure? Exactly. This is day 1, this is day 2, this is day 3, this is a 3-day procedure. So what is the use of inspissation? Two examples that you have to consider over here. There are two L's actually, and that is why we always give the mnemonic of ill for this. What is ill? That is inspissation is used for two L media. One of them is the LJ media. I hope you remember LJ media is used for tuberculosis detection, and the other one is Loeffler's serum slope, and that is used for Corynebacterium diphtheria. So the two L's are going to be sterilized by inspissation. There's actually another thing that comes in below 100, and that is going to be a water bath. So please remember, water bath is something where I have learned 60-60, or you can simply learn 60 degrees centigrade for 60 minutes, and that is what is going to be a water bath. So in very simple terms, it has to be below 100. That is there, and for 1 hour. So water bath में 1 hour में क्या रखते हैं? That is going to be the vaccines. Vaccines are usually kept in a water bath. Now let's come to at 100 degree. Do you remember I gave you a mnemonic called IPL BAT? So below 100 with inspissation and pasteurization. Now let's talk about what is A for at 100. At 100 you have B and T, that is boiling and tyndallization. Boiling, I'm sure everyone is knowing how to boil water. All that you have to do, देखो temperature तो 100 degree ही होगा. At 100 degree, boiling of water is roughly going to be for 15 minutes, right? But when you already had something at 100 degree, why would you need something else again at 100 degree? Why would you need that? And that is because only if you boil something once. देखो boiling में क्या था? You took a glass of water or a pan of water, you boiled it for 15 minutes and that's it. This way you did not kill spores, not killing the spores. So remember, this is the disadvantage that boiling has. That means I have to come up with tyndallization, which is going to definitely kill the spores. How will it kill it? Temperature the same, but now I have converted it into a 3-day procedure. So remember T20. What is T20? Another, everything has to revolve around cricket in this mnemonic. So what is T20? Temperature, I already know it is going to be at 100 degrees, but the timing is going to be 20 minutes, 20 minutes, 20 minutes. So this is also a 3-day process. That is day 1, day 2, day 3. It is 120, 120, 120. So remember, inspissation was 80-20, 80-20, 80-20. Tyndallization is 120, 120, 120, and that is going to kill the spores, and that is why it's better to boil it. Now we come to above 100 degree. A for A. Above 100 degree, we have autoclave. And autoclave, how much above 100 degree? Remember, 121 degree centigrade is going to be considered for 15 minutes for 15 psi. If you want to know what is psi, it is pound square inch. Basically, it's a unit of pressure. How much? It's actually the pressure cooker. I'll show you pictures. Look at this. These are all the pictures that you have for an autoclave. Different techniques. This one looks exactly like a pressure cooker. In pressure cooker, do you have pressure? Yes. So, देखो steam outlet, the whistle of a pressure cooker, everything is similar to a pressure cooker out here. So, this is the pressure cooker type. This is the autoclave which is long axis. It is vertical. Then this is the autoclave which is lying down. It is horizontal. This is the autoclave which is looking more or less like a washing machine. This is what is used in laboratories. This is the autoclave which is a very huge autoclave that is used for very large hospitals where a lot of samples come. So, these are the different kind of autoclaves. But one thing I understood was that whichever kind of autoclave that you're using, you have to follow 121 degrees centigrade for 15 minutes and 15 psi. That is the rule. However, guys, now this is something that I'm telling you for advanced exams. If you're targeting to kill prions, you will have to increase the temperature to 134 degrees centigrade, and you will, instead of 15 minutes, a dot laga do. 1.5 hours is the time that you will have to consider. So to kill the prions, the temperature is increased and the timing is also increased. That is what is autoclave. So what all do you keep inside this machine? You will always see the autoclave machine is seen in two places of your hospital. One is a path micro wala lab or lab related things, and one is the operation theatre. So why do I say that? That is because in an operation theatre, all the instruments, all the aprons, all the sutures, they are put in an autoclave. Which suture is not put in an autoclave? And that is going to be catgut. Catgut is extremely heat sensitive. Over here, you are going to have a very high temperature of 121 degrees centigrade. In that, if you put catgut, catgut is going to get destroyed. Elasticity gets destroyed. Never put catgut at such a high temperature. So all sutures except catgut are put here. All the culture media, so in a path lab or a micro lab, all the culture media except which two culture media? The L and L cannot be put over here. You know na, L and L kahan jayega except LG, LSS, because LG and LSS were going to be done by inspissation. All the other culture media put in an autoclave. So that is the microbiology lab use. And what is the pathology lab use? It is for sputum samples. So you can't just throw sputum samples in the sink or into any drainage. You will have to first autoclave it. Otherwise, that sputum will end up causing TB to the entire environmental and entire population. You don't want that. So first autoclave a sputum. It is your moral responsibility. Before you throw it into the sink or throw it into any biomedical waste, you autoclave it so that it is non-infectious. No one else in the environment or the hospital will get TB or infection out of it. So these are the things which are related to the OT. This is the thing which is related to the microbiology lab, and this is the usage in a pathology lab. That is why I said this machine will be somewhere hiding in a lab or will be put up in a OT. Now you will ask me what is the control. So now you remember the temperature is so high. I need a bacteria which can survive that much temperature and then get killed at a high temperature, and that is going to be Bacillus stero-thermophilus. The word only tells me, Bacillus stero-thermophilus tells me that it is capable of actually standing through long high temperatures. So thermophilus is what you're going to use for autoclave, and that definitely wraps up all of your the heat methodologies that you had to do: dry heat, moist below 100, at 100, above 100. Now let's come to the next one, and that is going to be filtration. So let me show you what are the different filters. Firstly, look at this picture. Look at this stainless steel photo and this actual photo. This is what we call as a seeds filter. We don't use it now. Why? Earlier they used to put water, like Nalla AquaGuard and RO systems. Earlier they used to use seeds filter. But why seeds filter not used nowadays? Because seeds filter is carcinogenic. If you put any water or solution through it, the solution that you get filter to hoga, but it will end up having some carcinogenic elements. Why? Because this contains asbestos, and I know from path and medicine that asbestos can cause cancers, lung cancers in humans. So I don't want to use it. Seeds filter is not used. It's just an MCQ. So आजकल क्या use होता है? You must have heard of these HEPA filters and ALPA filters. These are the different air purifiers. You must have seen a lot of air purifiers and water purifiers are being sold in the market. What is the usual pore size? The usual pore size is 0.22 microns. That is there for most of the very cut. Right? There is a variation from company to company, from air purifier to water purifier. But if I have to find an average of the pore size, the question that has come in previous year is 0.22 microns. So what all? This is all water purifier, air purifier to general public quota. What about a hospital and a lab? What all can you actually put through it? You will say that, ma'am, here one advantage is there. You are not using any heat. No heat, no increase in the temperature. So anything that is heat sensitive, like vaccines, antibiotics, toxins, sugar solutions, serum, these are all heat sensitive things. If you would have put it in autoclave at 121, you said destroy ho jata. So anything that is heat sensitive can definitely be put up over here. And what is the control? Now the pore size is so tiny. I need a tiny organism. So either you can use something called Brevendimonas diminuta, minute, reminds me of chota hoga, so it must be for filtration. And Serratia is also very, very small, so that is going to be for the filtration. These are the two controls that I use. So filtration is also done for us. What do we move on to next? We are lastly left with the radiation aspect, the last part of physical methods. How many type of radiation do you know of? If I go back to my basics of undergrad days, ionizing radiation and non-ionizing radiation. Give me some examples. Ionizing radiation has X-rays, it has X ka gamma bana do, so it is going to have gamma rays, and it is also going to have, this looks like a C, so it's going to have cosmic rays. Everyone repeat, it has X-rays, then you make a gamma out of it, so that is going to be gamma rays, and then you can also make it a C, that is going to be cosmic rays. Have you ever heard, if someone is like X-ray to karaya hoga, any X-ray, chest X-ray, orthopedic X-ray, does X-ray increase your body temperature? No, X-ray has no effect on your body temperature. So ionizing radiation, is it a hot radiation or a cold radiation? Common sense, it's going to be a cold radiation. It does not increase the temperature. Cold hai, but it is very, very highly penetrating. It is going to be high penetrating power, and they're going to kill all the spores. So remember, although it sounds cold, but look at it, it has a high penetrating power and is sporicidal. Versus what comes in non-ionizing radiation? All the things which are going to have a low penetrating power. I'll give you examples. We have UV rays, we have infrared rays. These are the things that are going to be. Now you must have heard UV rays, sunlight. Does it increase the temperature? Yes. Infrared rays, red color. Does it increase the temperature? Yes. So can I call it a kind of a hot radiation? Because these definitely increase the temperature, and this is what I'll be using for OT fumigation and biosafety cabinets. Biosafety cabinets are present in microbiology labs, the ones in which the microbiologist is working. That is a biosafety cabinet. And second, on OT fumigation also this is utilized. Versus where do I use these X-rays, gamma rays and cosmic rays, or ionizing radiation? The question that you definitely get, two questions. Cat gut sutures, because you were wondering since that time that all other sutures were put in an autoclave, if you remember. But I told you cat gut suture is not put in an autoclave. So where is the cat gut suture sterilized through? It is sterilized via the ionizing radiation. There is also the grafts. You must have heard of so many grafting techniques that are done. Kidney, bone marrow, so many grafts are done. They are also treated by ionizing radiation. So these are the two very important questions that you're going to get out here. Versus what is the control? Now simple. The way you write a P, is the same way you write an R. So Bacillus pumilus is the control for radiation. Bacillus pumilus is going to be for, the way you write a P is going to be for radiation. Radiation also done. That means I'm done with all the physical methods. It's a full wrap up. Now I can move on to the chemical methods. What all am I going to have in the chemical methods? Alcohols, aldehydes, halogens, phenols, plasma, and gas sterilization. Quickly, let's go through it. So first and foremost, let me start with alcohols. What all do you have to know under alcohol? Ethyl alcohol 70%, isopropyl alcohol 70%. Ethyl alcohol, I think everyone knows, is the spirit swab that we use. So the spirit swab in which you clean the skin or anything before you put a needle, that is a spirit swab. 70% ethyl alcohol is simple. Isopropyl alcohol is also 70%, and very, very important for a doctor because that is how you sterilize. Number one, your stethoscope, which you always forget to sterilize, and the second thing is a thermometer. Even a thermometer is going to be sterilized using it. So remember the stethoscope and the thermometer with isopropyl alcohol 70 to 80%. These were your alcohols. Now let's come to the aldehydes. You can see two aldehydes in front of you, formaldehyde and glutaraldehyde. Formaldehyde, I think everyone knows कि मैम अच्छा pathology से याद करें तो formalin is for all the specimens, isn't it? Formalin is going to be for all the pathology related specimens. This information I already have. Formalin is a liquid. Formalin is a liquid solution. However, if you're using formaldehyde as a gas, you will use it for doing OT fumigation, OT sterilization. Versus formaldehyde used in anthrax. What is that? So now we all know anthrax is bioterrorism agent. Anthrax is very, very deadly. Anthrax comes from animals. So you have to make sure that anthrax does not come from animals. So in the wool industry, animal wool, clothes will be made out of it. So many other things will be made out of it. So many people will be handling, the people who work in the wool industry. You want to save everyone from anthrax exposure. So what do? You do a procedure called duckering. What is duckering? That wool is treated with what agent? Wool is treated with formaldehyde. So that people who are handling the wool and using wool, they do not get bacillus anthracis infection. So formaldehyde is a gas for OT fumigation. Liquid ka to we know, that is why our eyes burn when we enter a path lab, because all the specimens are put inside formalin. And duckering is a process where I kill the spores, especially from animal wool. This is formaldehyde. Next I come to glutaraldehyde. This is not available as glutaraldehyde in the market. In the market it is available as Sidex. Every possible scope, laryngoscope, bronchoscope, endoscopes, all the scopes are put inside Sidex. Now let me tell you, all the scopes are put inside Sidex except a very recent and a very famous question, and that is going to be the arthroscopes. What is preferred for the arthroscopes is going to be the plasma sterilization, which I'm going to teach you in exactly 5 minutes. So plasma sterilization for the arthroscopes. But now they say that Sidex can have a problem. Before you start using Sidex, you need to activate it by alkalinization. That is one disadvantage that it has. And once you've activated the complex, you have to make sure that you use it also within 14 days. So that is a shelf life, and that is an extra step that you have for Sidex. But yeah, that is one thing which they really ask you. So now I can twist the question. All the scopes, bronchoscope, laryngoscope, endoscope, any scope, we are going to do with glutaraldehyde or Sidex, right? But don't get confused with arthroscopes, because that is plasma sterilization, and these are all being used in patients. Do not get confused with stethoscope. Stethoscope is something which is I'm doing it for patient to patient also, and also for myself. Stethoscope can I'm using isopropyl alcohol. These are alcohol and aldehydes. Now we come to the different kind of phenols. I think everyone has heard of phenol, which is a very famous antiseptic agent, 5%. We've heard in the market there are Crisols, Lysols. Lysol is a famous advertisement that comes on the television also, that is for floor disinfectants. We know these are all phenols. They ask me questions on modified phenol. So if I ask you, chlorhexidine and cetrimide का combination क्या होता है? That is known as Savlon. Whereas chlorxylenol is what? Chlorxylenol is going to be Dettol. How will I learn it? So simple. Dettol, chlorxylenol. Simple. Dettol is going to have chlorxylenol. Whereas Savlon. Savlon sounds like if the Sir wala stuff will come. So there's going to be cetrimide number 1. So there's going to be cetrimide, and Sir S also looks like a 6. So something to do with the 6 number. Hexidine. Chlorhexidine is going to come. So cetrimide and chlorhexidine is going to be Savlon, and chlorxylenol is going to be Dettol. So these are all the phenols that you have. We can make a line over here, because after this I have all the iodine and chlorine compounds coming. I think we all know that for cleaning the skin, either you use your tincture iodine or we use betadine. What is betadine coming under? It is a type of a povidone iodine. So whenever you want to clean the skin, either you use tincture iodine or you use povidone iodine or betadine. Whereas if you are dealing with some kind of a blood spill, which is the compound that you're going to use, you are going to use 1% sodium hypochlorite. Very famous question. You've seen it in your previous year papers. It's going to definitely come. Repeating, what do you use for blood spills? Small blood spills, large blood spills, again and again and repeating. It is 1% sodium hypochlorite. Very, very, very important. 10 star, 20 star, whatever you want to mark it. But please yaad kar lo. Okay, this is also done. So alcohol, aldehydes, phenols, halogens are done. Now I'm left with 2 lengthy ones: plasma sterilization and gas sterilization. So let me begin with plasma sterilization. Sterilization, do you know? But what is plasma? Plasma is a fourth, like we used to study in chemistry. What are the states of matter? Solid, liquid, gas. You to pata hai. Plasma is said to be the 4th state of matter, and that 4th state of matter is something that contains ions, electrons. You say, oh my God, how did you create plasma, man? We created it in a machine called STERAD. Now in the exam, STERAD word might be written on this machine, might not be written. So if it's not written, remember this white color box, Dabba machine is a STERAD. And STERAD is nothing but a machine for plasma sterilization. So why is it all closed from everywhere? I can't see what is happening inside it. Why is that so? Because I have to make sure that I have maintained a low temperature inside it, and I have maintained a vacuum situation. Only then I will create plasma. So कैसे plasma create होगा? You will say, ma'am, you will have to use a chemical called H2O2, and you will have to use UV photons. Then only. These are the 4 requisites that you have. क्या है? What all is inside this white color dabba? You have number 1, you have created an environment of a low temperature. Secondly, you have created a vacuum situation. Thirdly, you are going to utilize UV photons. And what is the chemical mainly from which plasma will be created? H2O2. These 4 things you have to know, because they are expected questions. So man, it's not complicated lag raha hai. But you have other techniques also which you just told me. There's also something which I teach you called gas sterilization. Iska advantage kya hai? Why is this better over gas sterilization? Because it is not having any toxic products. No toxicity and shorter time duration. It's going to take lesser time to perform. So where are we primarily going to use plasma sterilization? Mainly for the surgical instruments. And right now I told you one very important question. All the scopes were by Sidex, but one scope, that is arthroscope, that is going to be by plasma sterilization. In fact, if you read in a little bit more detail, the retroscopes are also now being considered by plasma sterilization only. What is the control? It is the same control that you used in autoclave. That is Bacillus stereothermophilus. That is about sterad. Remember, sterad matlab plasma sterilization. Very, very important, these 4 things that I told you. And I was comparing it with something known as gas sterilization. First question comes to me is, which gas are you using? I am using ethylene oxide gas. But why is this not as famous as plasma sterilization? Because this ethylene oxide has a lot of problems. It is inflammable, it is carcinogenic, it is explosive, it is irritant. Matlab har tarike ka problem hai. It will irritate you, it is explosive, it might catch fire, it might even be carcinogenic. So we are never going to use 100% of ethylene oxide. We are going to use 3 to 10% of ethylene oxide. Now they are saying 3% is good enough. You are going to mix it with other gases, with other inert gases. You are going to mix it so that you don't end up ethylene oxide catching fire and inflammable and irritation. So हो गया ना, toxicity है, irritant है, inflammable है, carcinogenic है, problem है ना. That is why I was saying plasma sterilization is better, because plasma sterilization was not having any kind of this irritation, toxicity, toxic products कुछ भी नहीं था. That is why it was better, and that is why I say that this gas sterilization has a lot of problems. But where are you using a gas sterilization? Mainly you are using it for the cardiopulmonary machines. You are using it for the cardiopulmonary machines. That is something important. So I'll make a tiny little heart and a lung over here so that you remember. What is the control? G for G. Gas sterilization ka control is Bacillus globi G. So you finished all the chemical methods, and I think it is the perfect time that you finish off all the controls also. Quickly, can you revise with me? What was the control that you had for a hot air oven? All the Bacillus, all the Clostridium. You could have Bacillus atrophius, Bacillus subtilis, and Clostridium tetani. For autoclave and plasma sterilization, your control is the same. That is Bacillus stereothermophilus. For filtration, you have to think of something chota pore, small size, minute. So Brevendimonas diminuta, because that is very minute, and something very, very small, that is Serratia, for small pores. For radiation, I told you P and R look the same, so it's going to be Bacillus pumilus. And for gas sterilization, I told you G for G, that is gas sterilization to the Bacillus globi G. So these are the ways that you learn all the controls that you had to know for the chapter. Before I end, I quickly want to make you go through something known as testing of a disinfectant. All this information I have told you, but if in case you end up asking me that, ma'am, I want to test a disinfectant, there is a disinfectant X, I want to find out, can I use it in my lab, can I use it in my hospital, in my clinic, is it a good disinfectant or not? What are the tests that you will do and tell me that haan, acha hai ya nahi hai? Test number 1, I will compare it with phenol. I compare with phenol because I know phenol is a decent disinfectant. If it is working, like for example, you take the average student of the class and you compare everyone's results with the average student. This is the pass fail. 40% is pass fail. If every student is getting 40%, matlab pass ho gaya. But that's not the best result. Na? You are comparing it with phenol. That is why you call it phenol coefficient test. You also call it Riedel-Walker test. But is this the best method? You just compare with someone who's getting a 40-50%. No. I want to compare it with someone who's getting a 90%. I want to compare the toppers of the class. So now I do, instead of Riedel-Walker, I do modified Riedel-Walker, and that is modified. Kuch modern naam sochna. Chick-Martin test. Chick is a modern name. Okay? So Chick-Martin test is modified Riedel-Walker test, and this is going to compare in the presence of yeast, soil. It will actually tell you the ground reality that if you have soil, if you have yeast, if you have fecal matter, is your disinfectant going to work? In the presence of yeast, in the presence of soil, or in other words, any kind of organic matter. If your disinfectant can work in that situation, it is a good disinfectant. So remember, there's something called Riedel-Walker test where you're just comparing disinfectant X with phenol, but that's not the best thing to do. The best thing to do is a modified Riedel-Walker test where you're comparing it in real ground reality. The third one and the fourth one, you might feel ma'am has written almost the same thing double, but read carefully. One is Kelsey-Sykes test, and one of them is Kelsey-Maurer test, and I have highlighted the center wala alphabet. Why? Because Kelsey-Sykes is K, it is the capacity test, and Kelsey-Maurer is going to be the in-use test. So what tells you the capacity and what tells you the usage? Now you know. So remember, Kelsey is fine. Kelsey-Sykes is going to tell me the capacity of a disinfectant, and Kelsey-Maurer is going to tell me the usage of a disinfectant. These are the 4 tests that I have for testing the efficacy, or testing whether my disinfectant is good to go or not. This was one table. The second table that I want all of you to know is, out of everything that I have taught you, what are the things that can kill the spores and what cannot? What are the sporicidal agents? E, F, G, H, ACT. Best way to learn it. What is EFGH? E, I think you remember, gas, ethylene oxide. F and G were the 2 aldehydes, formaldehyde, glutaraldehyde. H was a lot of things. H2O2, hot air oven, hypochlorite. I think this, hypochlorite has to kill the spores. Abhi to blood spills mein you were using it. Hot air oven was killing the spores. H2O2, where do you use H2O2? Ma'am, H2O2 is used in plasma sterilization. So one of the P's has to be plasma sterilization, of course. Okay, what is A? Of course, there will be one thing with A, and that is autoclave. What are the other things with P? One P, so I have done plasma sterilization. One P is peracetic acid, and one P is phthalic acid. Finally, T for tyndallization. So repeating once again, we have ethylene oxide, formaldehyde and glutaraldehyde. We have hypochlorite, hot air oven, and H2O2. H2O2 was also used in plasma sterilization, so one of the 3 P's is also done for you. APT, A for autoclave, P for plasma sterilization ho gaya, P for peracetic acid and phthalic acid, and T for tyndallization. These are the things that kill the spores. Finally, the last part of this entire classification and chapter that you have is the Spaulding's classification. What do you need to know under Spaulding's classification? It's a classification which says, okay, this is like a very, very critical device. This is a not so critical device. So this you need to, it's very critical. It's going inside your body. You need to use the best disinfectant. This is just the surface of the table. So iske liye dusting powder will only do. So you need to categorize things, what is very critical and what is not. And that is what Spaulding's classification does for you. What is a critical device? Something that is going to penetrate the body. So it could be a kind of a ventilator. It could be something that is going to go inside the body, or it could be some kind of a machine or an implant that you're going to make. So that comes under under a critical device, and for that you will use a high-level disinfectant. Semi-critical will be something that is coming in contact with the mucous membrane. Maybe some spopic method is going on. Anything coming in laryngoscope, bronchoscope, something coming in contact with the mucous membrane. For that also you will require a high-level disinfectant. Non-critical device means something that is just coming in contact with the skin. Now, for that, you don't need a high-level disinfectant. Intermediate or low-level disinfectant will be good to go for a non-critical device. What is non-critical? Something coming in contact with the skin. Non-critical device. Remember device, because if they say non-critical surfaces would be my table, my computer screen, any shelf. That's a surface. And for that, you can use a low-level disinfectant. So repeating, critical, semi-critical, and non-critical devices is alag, and non-critical surface is going to be alag. For critical and semi-critical, you use high-level disinfectants. For non-critical device, you can use intermediate or low level. For surfaces, you definitely go for low-level disinfectants. And this entire classification is referred to as the Spalding's classification. On that note, you definitely wrap up all of your general microbiology and you're good to go with the different systems now.